Good morning. We're going to call this meeting to order, but we're going to delay about 5 more minutes while everybody is getting settled, and we are waiting for 2 of our panel members who may be delayed. So we will be back on in about, let's say, in 5 minutes. Good morning. Now that our panel is assembled, I'd like to call this meeting to order. I want to welcome everybody to this meeting of the Scientific Review Panel on Toxic Air Contaminants. And I want to we are going to have viewers on the webcast watching. So welcome to them as well. A couple of administrative items for the people in the room. There are restrooms and drinking fountains outside the room to the left. Should there be a fire alarm, please exit down the stairs and proceed outside the building. Most importantly, the panel will really appreciate it if you set all your communicators to a silent mode. And now I'd like to just go around the table and have the panel members introduce themselves. Start with Alan.
Good morning. I'm Alan Buckpitt. I'm retired from the School of Veterinary Medicine at UC Davis.
I'm Stan Glantz. I'm a professor of medicine at UCSF.
I'm Kathy Hammond, professor of environmental health sciences at UC Berkeley School of Public Health and associate dean for academic affairs there.
I'm Court Anastasio. I'm a professor in the Department of Land, Air, and Water Resources at UC Davis.
Associate Professor in Microbiology, Pathology, Toxicology at USC and a member of the USC Norris Comprehensive Cancer Center.
I am Jesus Araujo, Associate Professor of Medicine and Environmental Health Sciences at UCLA.
Dr. Beate Ritz, Department of Epidemiology, Environmental Health Sciences and Neurology at UCLA, and a member of the Center for Occupational and Environmental Health at UCLA.
Paul Blanc, University of California, San Francisco, in the Department of Medicine, Chief of the Division of Occupational and Environmental Medicine.
Thank you. And I'm Michael Kleinman. I'm at University of California, Irvine, and I'm the chair of the panel. So this morning, there are two agenda items that we'll be going through. The first item will be a short briefing by the Air Resources Board to the panel about implementation of Assembly Bill 617. And then the second item will be the panel's review of the draft evaluation report on Chlorpyrifos from the Department of Pesticide Regulation. The Assembly Bill 617 requires that the Air Resources Board, among other things, develop a monitoring plan and emission reduction strategy for California's most polluted communities. The panel is one of several groups to be consulted in the implementation of this significant legislation signed by the governor last year. Today, Vernon Hughes, Chief of the Community Assessment Branch of the ARB's Office of Community Air Protection, will brief us on ARB's plans. Mr. Hughes, thank you very much for being here today, and I'll turn it over to you.
All right, thank you. Thank you very much for having us today. Again, we'll be giving an overview of the implementation status to date. As you all are probably aware, AB 617 passed in the summer, authored by Assembly Member Cristina Garcia, and it fundamentally transforms how we do community level planning for air quality, especially in communities that experience the greatest, highest cumulative exposure burdens. Specifically, the legislation sets out a number of planning framework elements, including community-level air monitoring, the development of a monitoring plan, a state strategy and community-specific emission reduction plans, an expedited schedule for the installation of the cleanest controls on facilities. So that goes back to backed, barked, T-backed type of controls. Enhanced requirements for the reporting of emissions, and increased penalty provisions for polluters, as well as makes available grant funds for communities and for early action items. And to implement the program, the California Air Resources Board has established the Community Air Protection Program and the Office of Community Air Protection, which I'm a branch chief there. And the legislation sets out ambitious implementation schedules to be achieved by October 1st, 2018. And that includes identifying impacted communities, establishing the criteria for air monitoring and local emission reduction plans, as well as developing statewide strategies for reducing emissions. It also gives air districts specific roles, so it's not only Air Resources Board implementing, the air districts play a very important role, especially in implementing the action plans called for in the bill. And to date, we've been working very closely with the air districts, with CAPCOA and air districts on working through, as partners, how we move forward with the implementation. We've also had a significant level of outreach to community groups, environmental groups, and regulated industries. So lots of meetings, and I'll touch on the scale or scope of outreach that we've done to date as well in a minute. But based on the outreach to date, we've put together 2 documents that provide staff's initial proposals on forming the program elements. The first one is called the Community Air Protection Program Framework document as a concept paper, and then also a process process and criteria for 2018 community selections. So part of the bill requires a process for identifying communities for action plans and monitoring, and that document covers the process for selecting the communities. The program framework is still in the early stages, so these documents it's really designed as a mechanism to seek continued feedback. Once we release them, they'll go out early February. Both the documents will go out for review and feedback. And we'll discuss these papers also in 3 summit meetings that we're going to be having, and all-day summits that we're going to be having in February. And those summits will be essentially a morning session that covers the broader program, and then breakout sessions that cover more specific elements of the programs. We plan to get into a fair amount of detail in those summits. AB 617 also prescribes a certain level of outreach in terms of consultation by requiring CARB to consult with specified stakeholders. Those stakeholders include this panel as one of the groups, air districts, Office of Environmental Health Hazard Assessment, or OEHA, environmental justice organizations, affected industries, and others. In response to those requirements for the consultation, CARB's put together a multi-stakeholder consultation group of more than 20 stakeholders, and we'll seek input from the consultation group through the consultation group. We'll seek consultation. Just because of the short time frames that we're under, that's an efficient way to bring all these folks together and get their input and feedback. Dr. Kleinman is one of the members of the consultation group, and it's going to be chaired by Dr. Balms, one of CARB's board members. And the first meeting of the group is scheduled for the end of this month. So we look forward to this committee's and the other members of the consultation group, their feedback on our program. As I mentioned, we've had a significant amount of outreach to date in the program. We've had of course, starting with the board meeting in October. Afterwards, we had a series of 4 informational meetings throughout the state on the program, and over 50 discussions with community residents, air districts, environmental justice organizations, and other stakeholders. Some of the themes that we heard during this public outreach process, they're certainly reflected in the concept paper and the papers that are going out early February. But we've heard an interest for a ground-up, community-based approach. The communities really want to be involved as these programs are put together, both for monitoring and the emission reduction programs. They're interested in steering committees, being involved in steering committees. Also a desire for increased monitoring, just to better understand what's happening, of course, at the neighborhood level, and establish criteria for setting up both the monitors in communities so that they're run in ways that the community certainly understands the results, and the results can be used for actions directly. They're interested in data transparency and on really understanding, again, being able to interpret the data themselves, if there are any sort of adjustments to the data for corroborating the data, say against federal reference methods, et cetera. They want to understand that and just be aware of how the data is processed. So a very transparent data stream is of interest. And they've mentioned the term, or coined the term, democratization of data. They're certainly interested too in being involved in deploying the networks based upon methodologies that can be standardized. They also want to focus on immediate, in terms of emission reduction programs, communities that the nature of the problem is well understood. there's been a lot of studies to date in certain communities, and they're very interested in getting action, where action can be taken and the data support those actions. Incentive funding programs, they're interested in programs that can help small businesses that are part of the community to support their efforts to reduce emissions. Some of those businesses have a hard time turning over older equipment, and so having a funding mechanism to facilitate turning over the equipment for those small businesses is of interest. And then, of course, along with the emission reduction strategies typical of statewide strategies and air districts, they're also interested in best practices, transportation strategies and routing of vehicles. So engaging local city and county governments as well. They're very interested in bringing not only state and local air districts, but also the city and county governments together. In terms of outreach moving forward, the community meetings, workshops, and all-day summits, they're going to continue in the future here. In February, the 22nd, we're going to have one of the summits that I mentioned earlier in Oakland, the 27th in Bakersfield, and the 28th in Riverside. So again, those summits will focus around the concept papers, having some very detailed discussion of the contents of the papers and looking for public feedback in those summits. In March, we have an informational board update where we're going to go over the status of the program and community selection, et cetera. Release of the draft framework document will come in May. So the concept papers and feedback we get from the public will feed into the broader, larger concept paper and framework document in May that'll pull all the information together to develop a statewide strategy and monitoring plan. And in June, we'll have workshops to workshop that larger document. And then in September, of course, we have the board meeting to consider adoption of the framework. That ends the status, I guess, to date on implementation. So thank you.
Sorry. Thank you very much, Mr. Hughes. I'd like to just ask if the panel has any questions. Well, I just want to mention that this whole idea of the community involvement, I think, is a lot of opportunity to both get a better understanding of what is happening in key communities. Quality and other issues will feed back to both the regulators better feeling for what is really important to people out there at the ground level. So I'm very happy that this program is stepping up, and I'm looking forward to participating. Kathy?
Just one quick thing. I appreciate this very much, the presentation very much, and the work you're doing. That's great. I'm just wondering if we could get some of those dates and some of that information in writing.
Sure. Yeah, I can send it to Jim Behrman.
Great.
The
Thank
list
you.
of dates. Sure.
All right. Well, again, thank you very much, Mr. Hughes.
You're very welcome. Thank you for having us.
I will now move on to our second agenda item, which is the evaluation as a toxic, getting an echo, as a toxic air contaminant. The Department of Pesticide Regulation distributed a draft evaluation of Chlorpyrifos, or it's easier to say CPF, as a toxic air contaminant to the panel on December 11th of 2017, and then followed it with an orientation presentation at our December 13th SRP meeting. DPR followed up that meeting by providing the panel with a bibliography of 308 references and the complete 2014 and 2016 U.S. EPA risk assessments. And if you look around the table, you will see that we're all burdened by many hundreds of pages of documentation. DPR staff have several individual panel members, and they'll be reporting back at this public meeting later. The next step in the evaluation of the next step in the evaluation of Chlorpyrifos as a toxic air contaminant is a peer review by the Scientific Review Panel, or SRP. The Toxic Air Contaminant Act is a statutory framework for identification, evaluation, and control of chemicals identified as toxic air contaminants. And the SRP today will review the DPR risk assessment to determine if it's seriously deficient based on a review of scientific data, the procedures, and the methods used to support the assessment's conclusions. Today we'll hear the technical presentation from DPR, then Oia will present their findings of the draft TAC doc evaluation. And this will be followed by a discussion of 6 charge questions that DPR specifically asked the panel to address. Because of the complexity of the CPF database and the risk assessment, which is, as you can imagine, goes on for in great detail, the panel may take more than this meeting, and in all likelihood will take more than one meeting to discuss all 6 charge questions and to decide whether the report is not seriously deficient, which is the requirement under the state law. If the panel decides that the report is not seriously deficient, we'll then submit written findings to the director of DPR, and he or she will, or he will decide, if Chlorpyrifos will be listed as a toxic air contaminant. If the panel finds serious deficiencies, then we will return the report to the DPR director for revision. As a reminder to everyone, including me, please speak directly into your microphones, and this is for the benefit of the court reporter and also for the listeners on the webcast. And with that, I'd like to turn the meeting over to Dr. Shelley Duteau, the branch chief for human health assessment at the Department of Pesticide Regulation, who will begin the presentation. And we're going to have a short recess while we're configuring the various communication devices. Okay, thank you very much for your patience, and I think we are now ready to begin the presentation.
Thank you, Chair Kleinman and members of the panel. Again, my name is Dr. Shelley Duteau. I'm the branch chief for human health assessment at the Department of Pesticide Regulation. We're very pleased to be here today to continue on our discussion of chlorpyrifos following the overview presentation at the December meeting. As you mentioned, Dr. Kleinman, we have a huge database. This is a big undertaking. So I realize there will be a lot of questions, but I did want to ask the chair prior to starting our presentation if you wanted to delay discussion and questions until after we talk and Oia gives their findings and then open the discussion, or did you want us to take questions during this presentation?
I'd like to make an alternative suggestion. I think it would be, I mean, if the panel doesn't want to do this, it's okay, but I think it would be useful to just go around the panel and hear what questions we have so that you can focus the presentation on the big issues. You know, I think that, because in reading through all of this, I mean, I had some fairly, you know, I think the issues are pretty clear, at least from my perspective. So, you know, there's a lot of issues in here that are probably not controversial. And, you know, to just have them know kind of what we're wondering about so they can be, you know, kind of focusing on those issues in the presentation. That would be my preference, if that's okay. I mean, I was one of the people who talked to the DPR people in advance, so they kind of know what I'm going to ask them anyway. But I mean, what do people think about that? Bad idea, good idea? Huh? Doesn't work? Okay, never mind.
Yeah, I think that because there is also not only the people on the panel, but there's a wider audience involved. I think it would be best to have everything on the table and then go from there. But in terms of, I think the best flow would be to have the DPR presentation followed by the Oia findings, which puts everything out there, and then we will start to focus on the charge questions.
Well, I would say that you should trust us that if we have a question that makes it impossible to understand what it is you're presenting, that we'll break in with those questions, because 120 slides later, for us to remember what the question was that we just lost you on is probably a disservice to everybody.
You'll be grateful then that we only have 50 slides.
Oh, he probably has 50 also.
20 slides.
Right, but you get the point.
Yes, thank you.
Well, I think, you know, in terms of this, if there are burning questions as the presentation comes up, I think it would be best to deal with those, you know, as they arise. But hopefully we'll still be able to get through this.
Great.
So I'll turn it over to you, Dr. Duteau.
Okay, then we'll go ahead and proceed. Joining me today, I have a distinguished group of colleagues joining me from the Department of Pesticide Regulation. Several of the scientists will be aiding in answering questions and clarifying, as well as some executive staff that are also here. So let me go ahead and introduce them. First of all is Dr. Marilyn Silva. She's the lead risk assessor for this document. Dr. Terrell Berry is the lead exposure assessor. Carolyn Lewis is a research scientist 3 with our department. Dr. Svetlana Koshlokova, our senior toxicologist in the risk assessment section. Dr. Eric Kwok, our senior toxicologist with the exposure assessment section. And also aiding us with additional questions if need be is Dr. Andrew Rubin, staff toxicologist. Mr. Randy Sagawa, special assistant. And Mr. Jesse Cuevas, our assistant director. Thank you all for being here. So today's presentation, I'm going to start by giving a background on pesticide toxic air contaminants. Because this panel hasn't had one of these come in front of them for about 8 years or so, it's good again to go over some of the information that we shared in December. Then I'll go over the steps in evaluating chlorpyrifos as a toxic air contaminant, or otherwise known as TAC. I might use that acronym throughout the presentation. We'll give an overview of DPR's assessment, which you received 278 pages plus additional documents. And start with discussion of questions from the panel and charge questions 1 or 2. And as Chair Kleinman said, if we get through the rest, then that will be great. So first of all, a background on pesticide toxic air contaminants, or pesticide TACs. DPR's authority is cited in the Food and Agricultural Code, and as mentioned before, requirements for the TACs are in sections 14021 and 14027. And it is defined much like a regular air contaminant in that it's an air pollutant that may cause or contribute to an illness, an increase in mortality or an increase in serious illness, which may pose a present or potential hazard to human health. In consultation with OIHA and ARB, DPR shall, let me spell those out for you, the Office of Environmental Health Hazard Assessment and the Air Resources Board. DPR, the Department of Pesticide Regulation, shall evaluate the potential hazards and health effects of pesticides that may be determined to be a TAC. And in consultation with OIHA, DPR prepares a report of health effects on the pesticide, which you have in front of you. This report addresses the availability and quality of the data on health effects, potency, mode of action, and other relevant biological factors, an estimate of the levels of exposure that may cause or contribute to adverse health effects, and the range of risks to humans resulting from current or anticipated exposures. To date, this is a timeline of the steps we've taken thus far. We first released a draft Chlorpyrifos Risk Assessment document, or RCD, in December 2015. Then we received technical comments from OIHA, ARB, and Dow AgroSciences. We evaluated and assessed chlorpyrifos as a toxic air contaminant based on this 2015 draft. We incorporated pertinent technical comments, and we included expanded analyses of new and additional data, including developmental and cancer epidemiology, in vitro and in vivo results, human exposure and illnesses, pesticide use, mechanism of the pesticide, Pbpkpd modeling, air drift modeling, and so on and so forth. Then in August 2017, we released our revised draft evaluation of chlorpyrifos as a toxic air contaminant. And we also released formal responses to the technical comments we received from OIHA, ARB, and Dow. In September, just 4 months ago, we presented a draft evaluation overview to our Pesticide Registration and Evaluation Committee, and we opened a 45-day comment period. During that time, the public comment period and the months following, we released a revised draft of the evaluation of chlorpyrifos as a toxic air contaminant. It now incorporates pertinent technical comments received during the public comment period and an expanded analysis of new and additional data, including developmental neurotoxicity in vivo results in animals. We also released responses to the technical public comments and additional comments we received from Dow. And we distributed the latest evaluation with all of those comments to this panel. We received, as did you, OIHA's findings in December. But then we followed up with an orientation presentation on December 13th. And we followed up with the panel with additional supporting materials that were requested. On the 17th, we released our response to OIHA's findings. And all of these documents that I mentioned are available online. So here we are at the first SRP meeting to review the data. And as Chair Kleinman mentioned, the panel shall review the scientific data on which the report is based, the scientific procedures and methods used, to support the data and the conclusions and assessments on which the report is based, and determine if the report is seriously deficient. So, on to the evaluation of chlorpyrifos as a toxic air contaminant. So chlorpyrifos is a very old chemical. We have data going back, I think one of the earliest reports we saw was 1948. It is a chlorinated organophosphorus ester, and it's manufactured by Dow as an insecticide, acaricide, and miticide, and the structure is shown here. It was first registered in 1965, meaning that there was plenty of data on its R&D prior to that. In December 2000, EPA reached an agreement to halt manufacture of chlorpyrifos for nearly all residential uses. And in March 2001, EPA registration was canceled for indoor residential products, except those that are containerized baits and child-resistant packaging. Outdoor residential products were canceled except for fire ant mound treatment by licensed applicators or mosquito control by public health agencies. And in December 2002, all retail sales were stopped. However, there's currently a large amount of agricultural uses in the United States that are registered by EPA, including on fruits, vegetables, tree nuts, and grain crops. In California specifically, we have 48 products that are actively registered. And the major uses in California also mirror those of the United States, including nut trees, fruits and vegetables, and grain crops. We also have several non-production agricultural uses allowed in this state, including golf course turf, industrial sites, greenhouse and nursery production, seed treatments, sod farms, and wood products. We also allow it to be used for cattle ear tags, again, roach bait and child-proof containers in homes and sewer manholes, for fire ant control, and again, for public health control of mosquitoes. We also have additional restrictions than EPA does. So we have it listed as a California restricted material. The major geographic use areas in California include the Central Valley, the Central Coast, and Imperial County. It's used year-round, but peak use is usually in the summertime, depending on weather. Allowed applications include aerial, air blast, ground boom, chemigation, and others. So this is the total pounds of chlorpyrifos used in California agricultural production since 2011. You'll see that the draft number here for 2016, which needs to be finalized in our PUR report, our pesticide use registry database report. So 2016 is a draft number, but you can see a trend in downward use of chlorpyrifos across the state. For human illnesses and exposure reports, California Department of Pesticide Regulation has a robust program, and they track these reported incidents. From 2004 to 2014, there were 246 cases of pesticide exposure from 84 episodes involving chlorpyrifos. And a majority of those cases were either due to drift, residue exposure, or ingestion, which was largely intentional, unfortunately. Only 5% of ingestion was accidental. No, ingestion, the majority of cases includes ingestion. Only 5%, you're absolutely right. I apologize. 5% of the majority of cases was ingestion, and it was largely accidental. But there are cases of intentional ingestion as well for suicide, attempted suicide.
I'm sorry, I don't understand what you just said.
Okay.
Could you say that again?
Okay, thank you. I will clarify. Ingestion cases made up 5% of the total cases. Of those ingestion cases, they were largely accidental. Usually that comes in the case of a pesticide being poured into a non-labeled container, sometimes a water bottle or something else. But there are cases of intentional ingestion as well.
And then I have another question, just a clarification on what you're talking about here. You keep calling these cases of exposure?
Yes.
Are they cases of illnesses or
There's a definition that our worker health and safety branch uses to define what a case, an illness report, and an incident and an episode is. I don't have those definitions right with me, but I can ask Randy or Jesse if they'd like to clarify.
Good morning. I'm Randy Sagawa, Special Advisor with DPR. So yes, DPR has a pesticide illness surveillance program, has had one for several decades now, and so we track illnesses due to pesticides. And so in this case, we are talking about actual acute illnesses, and that's what we're referring to when we say cases.
Okay, so I think that that's another error on the slide. It's not 246 cases of exposure, but of reported illness. Is that correct?
Correct.
I think that's an important distinction.
So each case is an individual or a group?
Correct. And so there are in this case, there are 246 individuals that became ill due to chlorpyrifos exposure that were reported. And those 246 individuals were exposed in 84 different incidents or episodes.
And then following that, when you say majority of cases, those add up to the 88%, and those are just bystanders?
No, illnesses are reported both for bystanders as well as occupational exposure.
So, if I'm jumping ahead, stop me, but how many of these cases were bystander and how many were workers?
If I hit the next button,
then I'll wait. Let's just don't
it's
go
right
forward.
there.
Oh, okay. So.
And bystanders, Randy, if you could add a definition of bystanders, because it's not just residential bystanders, occupational bystanders.
Yeah, and so when we refer to occupational exposures, it's the people who are handling and applying the pesticide.
So you had relatively few, only 12%, less than 12% of the cases that were reported. But again, the cases are not cases of exposure, they're cases of reported illness.
Correct.
I just want to point out for those of you who are less familiar with the pesticide illness reporting system, it's a passive surveillance system that relies on reports being filed with the county ag commissioners, usually or county health commissioners. There's been a component expanded wherein cases are solicited from the California Poison Control System, but that does not span this entire period. I do not believe, I could be wrong, about how long you've had that arrangement with
Yeah, I'm not sure
California.
either. Yeah, but you're correct that we have been consulting with Poison Control to get their data.
Right.
We also consult with worker compensation to get their information as well. And so we try to get as
I
complete
understand
amount of
that,
data
but
as possible.
it's an incomplete surveillance system. So one of the things that would be important, since the inconsistencies in the system are fairly stable over time, would be to get a sense of the temporal trends in the 10-year period in question. So is there, the next slide have the temporal trends perhaps? Good, thank you.
Okay, so again, here are the temporal cases plotted as cases and episodes from 2004 to 2014. And much like the plot of chlorpyrifos use statewide, these reported cases have generally trended down over the same amount of time. And the next plot is illnesses caused by agricultural use. And in blue is the non-occupational or the non-handler cases, illnesses, and in the green are the occupational. And again, there's a trend over time of less of these with less chlorpyrifos use across the state.
I have a question on that. I thought you said more than 88% were non-occupational. That doesn't look like that's true, looking at that figure.
I think that,
Kathy, you have to understand, if I'm a farm worker and the plane flies over me and sprays me with chlorpyrifos and I get ill and go to intensive care, that's not occupational in their system, that's bystander. I have to be the pilot applying the material to be ill to be occupational, or the person formulating it.
Okay, so
So
these,
this, some of these people are, you know, the
So
kid playing at the school, but some of them are the farm workers, but they're not occupational in this system. Randy, correct me if I'm wrong on that.
But I think that it's like we have two different definitions then on this slide and 2 slides back that are confusing. I hear what you're saying, Paul, I get it. But,
and I apologize for any confusion. This report and the graphics from it are generated from a report from our Worker Health and Safety branch, and it is referred to as one of the first references in the back of your risk assessment document. And it's available online, and if the committee would like to have copies of it, we'd be happy to make copies during the lunch break and distribute them.
I think that would be a good idea.
Okay, we will do so.
I do think just that the point Kathy's raising, if you just look at the slide and you just add up the occupational numbers, that looks like that's more than 12%.
Agreed, and it does look confusing, and I wish I could explain the numbers more, but it was generated in a different branch.
yeah, if one way that could perhaps help on this is, I hear Paul's point that under non-occupational, under occupational here are people that are bystanders in the other, in the 88%.
Occupational bystanders.
So, but you could perhaps make a third color here, that were bystanders, people who were occupationally exposed but not.
Sure, that I absolutely understand what you're saying, Dr. Hammond.
Yeah. Just
Occupational bystanders, residential bystanders, and then occupational ag handler exposures. Those 3 colors would make a tremendous difference in this graph. And knowing that would clarify, we will certainly move forward with a suggestion to the Worker Health and Safety branch for their subsequent reports that come out annually to perhaps make the graphics that way.
And also, I would be interested in a graph that breaks that into different age groups.
Different age groups.
How many of these illnesses are among children, for instance?
Okay, so that would be the residential bystander. Got it.
Unless they're children in the fields.
Wow, Cal OSHA might have an issue with that. Although I realize that there are restrictions were lifted on the federal level for who can use pesticides, age-wise. I think that goes to down to age 16, or that was somehow changed. So we'll have some complicated numbers coming out in next year's report, but I appreciate the chance to try and clarify. I appreciate Randy coming up, and we will certainly make an effort as a department to come out with a more clarified assessment of the data for future year reports.
And I appreciate your efforts there too. Thank you.
Thank you. Okay, moving on to the fun stuff, symptomology. Acute poisoning of chlorpyrifos can result in human deaths. Doses generally over 300 milligrams per kilograms in humans would be required to cause death, but it also can result in unconsciousness, convulsions, cyanosis, and uncontrolled urination. Lower doses, or those less than 300 milligrams per kilogram, can result in hypersalivation, respiratory distress, muscle tremors, and the general range of symptoms seen with other organophosphate poisoning. For chronic toxicity in workers, workers who have reported higher exposures reported impaired memory, some speech difficulties as well, but there were no consistent reports of effect in workers exposed to lower levels. And this is from Albers et al., 2004. So moving on to the toxicology profile. As I mentioned, there's chlorpyrifos has a classical mode of classical target a toxicity associated with being an organophosphate in that it binds and inhibits the enzyme acetylcholinesterase in both insects and mammals. The effect on insects is why it is used in this state and elsewhere as an insecticide. In mammals, Chlorpyrifos results in the accumulation of the neurotransmitter acetylcholine, and this results in excessive stimulation of the cholinergic pathways in the central and peripheral nervous systems, and it requires metabolic activation of Chlorpyrifos to the oxone to inhibit this cholinesterase activity. Acetylcholinesterase hydrolyzes acetylcholine in some non-neuronal cells, such as red blood cells. And RBC, or red blood cell inhibition, is commonly used as a surrogate of the inhibition in other target tissues, including brain. The threshold dose for red blood cell acetylcholinesterase is approximately 1 milligram per kilogram per day, including for immature organisms, and these are in a mammalian system. And this number will come up several times throughout our discussions today with you. In terms of
I just have a question.
Yes.
So when you say mammalian systems, is that all mammals, including humans, or are you talking about, you know, rats and people are different?
Mammalian, so it spans the kingdom of mammals. In terms of toxicokinetics, we'll break this up into absorption first. Oral absorption is complete, approximately 70 to 90% in rats and humans. Dermal absorption is much lower, though, at only 3 to 10%, as found in urinary metabolites. There's also evidence that through inhalation absorption that acetylcholinesterase can be inhibited. In terms of distribution, the highest levels of Chlorpyrifos are found in fat. It also tends to bind to plasma proteins. It's also been detected in rat and human milk, and there's also evidence of transplacental transfer. Metabolism is generally handled by liver cytochrome P450 enzymes. Chlorpyrifos can be oxidatively desulfurated to the oxone, and it can be dearylated into 3,5,6-trichloro-2-pyridinol, or TCPY, as well as diethyl thiophosphate, or DETP. And it can be hydrolyzed into TCPY and diethyl phosphate, or DEP. Elimination is generally rapid in mammalian systems. The biological half-life in humans is between 10 and 27 hours. Urine is the main route of elimination, and you will find TCPY as well as the other two metabolites mentioned, and also glucuronide and sulfite conjugates in urine. And urinary TCPY is commonly used in human biomonitoring studies. The targets of toxicity have been looked at extensively in multiple systems across animal models, and this was actually a requirement of FIF registration. So we have multiple data volumes showing the results from the various targets of toxicity. So first, in terms of developmental and reproductive toxicity, there's no evidence that Chlorpyrifos is a teratogen or that affects reproduction. I did come across one paper recently that showed that 35 milligrams per kilogram per day in a mouse model affected spermatozoa, but that's significantly above the level that causes acetylcholinesterase inhibition. And no fetal toxicity was observed without the presence of maternal toxicity, or to say another way, as the slide bullet says, fetal toxicity is only observed in the presence of maternal toxicity. We're talking overt toxicity in the pups. Immunotoxicity doesn't seem to be a target for Chlorpyrifos. And there are studies for genotoxic effects. Those are mostly negative. However, DNA damage has been shown in assays with yeast and bacteria, as well as cell culture from treated laboratory animals.
let me break in here just so
Sure.
I'm clear about your terminology. By inference, I'm taking it that you exclude from developmental toxicity on this slide, developmental neurotoxicity.
Absolutely, yes.
So I think that's rather important to say, and the slide is actually quite misleading in a very critical and crucial way. Not that you intended it to be, but I think we have to call that out right now, that in future slides you're going to get to neurotoxicity, but most of us on this panel would consider developmental neurotoxicity as a developmental subset of developmental toxicity. So if I were editing your slides, I'd put a big parenthesis, say, except for, you know.
Thank you for the clarification. You're absolutely correct. In this case, it would be ossification, cleft palate, those sort of gross malformations that we look for. But you're absolutely right. So Svetlana is reminding me that how I'm going through this is actually following the table of contents of the risk assessment document that you received. But you're absolutely right. We don't want to mislead by implying in any stretch of the imagination that chlorpyrifos does not potentially affect neurodevelopment. In terms of carcinogenicity, in animal studies, there has been no evidence of tumors in chronic feeding studies of rats and mice. In humans, there have been reported associations between chlorpyrifos use and non-Hodgkin's lymphoma, lung and rectal cancer in pesticide applicators and farmers. This is largely from the agricultural health study. These associations are based on relatively small numbers of cases, large numbers of controls though, and concomitant exposure to other chemicals happen in many numbers of these cases. The exposure is based on questionnaires, either as recall with occupational, it's not handlers, it's actually farmers, or questionnaires with the family members if those farmers had been deceased. So it's a recall on what they might have used when they were farming. According to the US EPA, chlorpyrifos is not likely to be carcinogenic in humans, and that's based on the lack of evidence of carcinogenicity in animal studies and the absence of mutagenicity. Okay, neurodevelopment toxicity. In animals first, there are many studies where females were dosed throughout pregnancy and through lactation, or that the pups were dosed after weaning in repeated dosing. So again, the females being dosed throughout pregnancy and into lactation so that the pups were exposed in utero and through milk, or the pups, when they were a certain age, were then dosed after they were weaned, and it was repeated dosing, generally through gavage. The animals were evaluated for their motor activity, auditory startle response, spatial orientation, social behavior, cognition, anxiety, in young pups as they matured. We have many results in our December 2015 and 2017 draft risk assessments, and here's a summary of some of those findings. That developmental neurotoxicity occurs at doses that do not alter pregnancy or the general health of the offspring. And there's evidence of long-lasting impairment on locomotor activity, deficits in cognitive function and social interaction at doses equivalent to the threshold for cholinesterase inhibition, or 1 milligram per kilogram per day. There's also evidence of a decline in anxiety shortly after weaning associated with doses below those that inhibit brain cholinesterase, or levels approximately 0.5 milligrams per kilogram per day. In addition, chlorpyrifos has been shown to inhibit neuronal growth in vitro at concentrations well below those that cause acetylcholinesterase inhibition. And Dave Eaton did a nice review of this in 2008.
Can you just clarify one usage of terminology here? When you use the term at the threshold of acetylcholinesterase inhibition, do you mean at the threshold of 10% inhibition? Or do you
It is
mean at
10%
the
in the
threshold
animals,
of
correct.
0 inhibition?
It's not zeroed out, it's just a 10% decrease.
Yeah, okay. So that means it's important to be clear about that.
Correct.
Since the 10% is what you use for other endpoints.
Right.
Is that correct?
Correct.
So therefore, the interpretation of this is a mixed bag where some of the effects are at the 10% threshold, but some of them are well below those that the in vitro are well below those at which any inhibition whatsoever can be detected.
Right, and the second bullet from the bottom, the anxiety effect happens without the cholinesterase inhibition at doses that don't cause cholinesterase inhibition.
Not even 3%, just 0%. Is that correct?
So many of the new studies that measure those low-level effects, they do not concurrently measure cholinesterase in their assays. So we are going by the general threshold for cholinesterase inhibition.
But that wasn't exactly my question. My question was, yes, it's below the 10%. Is it 1% or 0.1% inhibition or no inhibition that could possibly be measured?
So in general, for cholinesterase inhibition, 10% is considered the limit of detection, the detection limit.
I see. Okay, thanks. That's helpful.
I think it would be very difficult to measure 1 or 3% inhibition. I mean, you're measuring small differences in large numbers.
I suppose it's a statistical question. If you had enough samples, you could certainly measure 3% inhibition reliably if you had enough observations. But I don't know what the variability is, but assuming you had enough observations.
So in 2006 or 8, EUCPA and NTP conducted a large data set analysis of cholinesterase inhibition in brain and RBC in the red blood cell, and the 10% was determined as the threshold for detection. Not the threshold, the detection limit.
And just for my clarification, meaning that it's difficult to quantitate levels of inhibition less than 10%. So between 1 and 9%, it's very difficult to quantify analytically, correct? Yeah, and enzymatic issues are hard to quantify at times.
And
Do you happen to remember what the number was? How many analysis they did in that NTP study?
We'll get back to you on this. I recall 100 data set.
and
I think it's for brain cholinesterase. I don't recall. I don't remember the RBC.
another question on that? Oh, yeah.
We'll get back.
So just another quick question. This is just my lack of knowledge in toxicology. Is there a linear relationship between Chlorpyrifos and the inhibition, or is that sigmoidal, or what's the relationship there? From say 10% inhibition going up, is that a linear relationship? And what concentration gives you 10% inhibition?
1 milligram per kilogram per day is generally the dose that causes 10% inhibition.
10% inhibition? Okay, thank you.
So again, we will follow up with the panel on the NTP report. Additional questions before moving off this slide? So we have expanded and looked further into additional data on neurodevelopment and toxicity in animals. And those results are shown in our 2017 draft, which is the one that you have. And we will also follow up with subsequent drafts, any additional data and findings that we have. Most of these additional data were found in abstracts of presentations or published papers from 2014 to 2017. And those reported developmental neurotoxicity, that is in pups, or neurotoxicity in adults at low wells of 0.1 to 0.5 milligrams per kilograms per day. So that would be approximately 1 order of magnitude lower than the amount that causes 10% inhibition in acetylcholinesterase. So there were 3 domains that were studied in this, this small group of research from 2014 to 2017. And the domains that were studied are cognition, behavior, and motor activity, again, in animal models. There were 2 rodent species tested, both rats and mice. And the treatment was largely by gavage for dams during pregnancy and lactation or pups, or indirect exposure to the pups in utero and through lactation, or subcutaneous treatment of adults. So that's simply the neurotoxicity and not developmental toxicity. Exposure was either fully gestational or only postnatal or both, and it consisted of either single or repeated doses. And this is a table of some of these results, including Carr et al. 2015, and Professor Carr was on the SAP panel that was asked to talk about biomarkers in the April 2016 meeting with EPA about chlorpyrifos. Silva et al. Is one of the most recent papers, as well as Gomez Jimenez. And Lee et al. Also has data on chlorpyrifos and carbamates. And Mueller was, I think, one of the only papers that, the new papers that we looked at adults only, and that was the subcutaneous testing. So just in terms of a summary table here, you're given a large slide of it, but if I could point your attention to when you have an opportunity to look at in the risk assessment document, tables 12, 13, 15, and 16 have additional data besides these to look at. So in general, looking at all of the new data and the older data, behavior and cognition are relatively difficult to assess and quantify in animal models. You can't ask the mouse how they're feeling. It's really hard. But it's complicated in that some effects don't show a dose response. It's really hard to do a dose response on a startle effect. How do you quantify that? And others are equivocal. Sometimes the response goes up, sometimes it goes down. Also, of course, you would think that the neuronal pathways involved in regulation of motor and behavioral behaviors are not all known, and I'm sure that they somehow work with each other. We haven't completely elucidated all of those pathways. And as Svetlana, Dr. Kloshilokova was mentioning, cholinesterase activity or inhibition was generally not concurrently measured in these studies, so it's hard to know what the level of inhibition was occurring with the other effects that were being measured in terms of motor, startle, reflex, et cetera. And another complicating factor with published literature as opposed to contract laboratory reports we receive through the FIFRA requirements is that individual animal data are generally not available. Sometimes we do go back to the researchers and ask for individual data to be able to perform further analysis, but for these particular 6 or 7 publications, we weren't able to get individual data to perform further analysis.
Excuse me.
Yes.
Were you not able because of time, because these papers just came out and it just hasn't been time? Or have you asked them and they've said no?
Largely, it's been a matter of time, but I'll defer to Marilyn. Have you had a chance to? Yeah, we haven't. There's occasionally researchers are happy to give us the information. We have a recent call out to a researcher in Sweden for carbaryl who was willing to share the raw data with us. It's not always the case.
So if I'm hearing you correctly, you sometimes do ask for those data and then do a further analysis.
Yes.
So it would seem I certainly understand this is 2017 data, just a month, So is that something you have asked for, or you're going to ask for, so you could include them in the next iteration?
So we did ask for raw data. One of the groups, this was from Rio and Chlorpyrifos. By Lee, yeah, and they did. We did receive the data. No. Can you go back to the 2015 paper, the brain? Yeah.
That is the one paper we received individually.
Yeah. So we requested this for carbaryl, not for chlorpyrifos, because that's when we started. That's why we started looking at this paper. But we can get the chlorpyrifos data if they're willing to share.
And Dr. Silva, did you mention that you wanted to try to get the Mueller data as well? No. She said no, that she hasn't requested those data. But if we do get individual data and can do our own analysis independent of the manuscript, we will certainly do so for either a subsequent draft of this document or the final draft. Okay, moving on to neurodevelopmental epidemiology. There are several ongoing prospective cohort studies and multiple observational studies that have investigated the associations between markers of chlorpyrifos exposure and effects on neurodevelopment, learning, and behavior in humans. Some of the studies included biomarkers of exposure, such as chlorpyrifos measured in plasma, TCPY, or nonspecific OP metabolites in maternal or child urine. One of these studies was conducted by the Columbia Center for Children's Environmental Health at the University of Columbia. It's also known as the CCCEH cohort, so if you see that acronym, that's referring to the Columbia cohort. In this study, they quantified chlorpyrifos in maternal or cord blood plasma around the time of birth. They also are unique in that they did personal air sampling for the mothers in and around the time of 1 month prior to birth. And in this study, they evaluated associations between plasma concentrations of chlorpyrifos around the time of birth and attention problems, attention deficit and hyperactivity disorders, pervasive developmental disorder, working memory, and full-scale IQ in the offspring of the mothers. The Columbia study is actually not the only study that tried to quantify maternal or cord blood plasma levels of chlorpyrifos at the time of birth. Digging into it further, we found several studies that have done the same thing. So the study from Columbia, Wyatt et al. 2003, that most people refer to is highlighted up in the light blue at the top of the table. And in addition, there have been other studies from 2010, 2012, and 2015 and 17 that have also done the same type of quantification of the parent compound in cord blood plasma around the time of birth. They all differed in the number of samples, their limit of quantitation or detection, the median level of chlorpyrifos detected and their methodologies. I realize this is the first time you've seen this data. This is not in the draft. This is a brand new assessment. So I'd like to show or point out some of the information here that might be interesting to you. First of all, there was a difficulty in finding samples that were above the limit of detection. In restating that, it would be to say that chlorpyrifos was difficult to detect in samples. The rate was less than 68%, and some studies only 1 or 2% of the samples were above the limit of detection or quantitation. And that doesn't mean that the sensitivity of the analysis was difficult. It's just chlorpyrifos is difficult to find in the blood, and it's rather reactive, and it has a pretty short half-life. The limit of detection or quantitation ranged from 0.001 parts per billion, in other words, 1 part per trillion, up to 21 parts per billion. So several orders of magnitude between the analytical limits of detection and quantitation. The medians also ranged as well as significantly. Some of these studies did not report median levels and simply reported a range. So for instance, the median range for Wyatt et al., the Columbia study, was 0.026 parts per billion, or 2.6 parts per trillion. However, if I may point out in their methodology, the last column, their standard curve was between 21 and 6,400 parts per billion, meaning that the low end of the standard curve was 3 orders of magnitude higher than the median of the samples that they tested. So it's difficult to know with accuracy if those measurements were precise or accurate. I also would like to note that the recovery of chlorpyrifos from those samples was only 18 to 21%, meaning that if exposure was attributed to these samples, it was underestimated. By how much, we're not quite sure, since the recovery was so low. In other studies, the limit of detection, such as in Netta et al. 2010, the next line, the limit of detection was actually above the highest the range of the samples they tested. So it's difficult to have confidence in a number which is supposed to be the maximum concentration of chlorpyrifos in blood when the limit of detection was actually higher than that number. And in Huen et al. 2012, for the Chamacos study, that was also the case, that the median levels of both the cord and maternal plasma sampled were below the limit of detection. And in that paper, the authors note that they do not have confidence in using the results from those studies to do any further analyses. So I'll let this information percolate. Again, it was we did this brand new between the time that we released this information to you and this meeting, and we can certainly add this as additional information to the revised risk assessments that will be coming out. Yes, Dr.
So
Ritz.
this is the parent compound, not the oxone or TCPY.
Right. This is parent.
Right. So we would expect very little of that, right?
Actually, what I understand, and perhaps Dr. Kwok can weigh in, is that only about 40% of chlorpyrifos goes to the oxone. The rest goes through other metabolic pathways. So if you're measuring the oxone, you're actually measuring even less of the component. So Eric, would you like to clarify anything further on that?
I don't have anything to add. It's At this point, I'm not very familiar with the branching ratio and how actually different pathway. If you have a same parent compound, that you have multiple pathway on it to be able to accurately characterize that, you will actually need to consider the kinetics of each pathway and then to determine actual branching ratios. I haven't actually done this analysis. I haven't seen anybody actually done this type of analysis. So at this point, I really have nothing to add.
Kathy?
Thank you very much for bringing this new material here, and I share your concerns about the limits of detection and some of that. Is there information that would help us interpret this? I know there is, I'm sure, that relates the plasma concentration to percent inhibition?
That's a really good question. In fact, your colleague, Dr. Isaac Pessa, asked the same question at the April 26 Scientific Advisory Panel meeting.
Oh,
at
and did they get an
USEPA.
answer?
And he thought with a part per trillion quantitation in plasma that would range to even a part per quadrillion, or that would be pico to femto levels of molecules hitting the target. And I know there's a direct quote in the transcript from that meeting, but from my recall, I believe he said that if chlorpyrifos actually acted at that level, at that concentration, it would be more potent than the most potent pharmaceuticals that we've engineered to this point. But to be able to quantify what this means in terms of inhibition, it's very difficult to do because, like some of the animal studies, acetylcholinesterase was not measured at the time these samples were taken.
Oh, darn. Right, okay. And so no one has, not necessarily even in these studies, but in other studies, no one has actually measured both the plasma level of chlorpyrifos and the percent inhibition.
Not in terms of developmental neurotoxicity.
Well, I'm just asking, even just at a higher end of the
I
dose
actually
response.
do believe that there are some occupational studies and maybe even agricultural health study, perhaps, and I'm not sure if
I guess it'd be worthwhile piecing together what data are there and just
Sure.
seeing what the picture looks like.
That's a
Might
very
be
good
occupational
question.
here and something else here, and just seeing how they could come together.
Right. And I know that there are occupational monitoring programs where they have measured it. And so that would be very interesting to look at, indeed.
Okay. And then, stop me again if I'm jumping the gun, sorry. But do we have a time period in gestation that we think is most important for neurodevelopmental toxicity? I thought it was earlier. There's some,
right.
And we should all keep in mind that this exposure determination is made at birth, right?
Correct.
And if the mother's been in, if we have something that has a half-life of 27 hours, and a mother's probably been kind of more removed from most of the exposures, that it may be that this is an underestimate. I don't know.
Right. And
it's really hard to know what's happening there.
And you're absolutely right. Several of the studies in their discussions have talked about what is that critical window vis-a-vis an organophosphate or chlorpyrifos specifically in terms of neurodevelopment. For most chemicals, as you know, it would be in the first trimester into the second trimester where you have the neuronal tube development, et cetera. And then most of the growth and development happening in the third trimester. The difficulty that the studies have pointed out is there's inconsistencies with what the plasma level at birth are and the quantitation of urinary metabolites. Those don't always seem to line up in some of these studies. Interestingly, with the Columbia study, we also had personal air sampling of the mothers, 48-hour time-weighted averages, and they were not correlated well with the blood samples at birth either. So when we move from a measurement at birth and trying to figure out, is that similar to the exposure over a 9-month period, we have very little data to allow us to figure that out, unfortunately.
The Columbia population were inner city, right?
Right.
So they probably don't have agriculture, but they were also near the turn of the century, so they could have had the residential pesticides.
Right, and it's interesting that that study, as well as the home study in New Jersey, that cohort spanned the time of indoor use and banned or restricted indoor use. And so you have, I believe there might be a discussion in that in our current risk assessment where it talks about the enrollees prior to the ban have quantifiable measures of various things, urinary metabolites, et cetera. But after that ban, it's virtually impossible to detect chlorpyrifos either in plasma samples or in urinary samples as well.
So in addition to these biomarkers, I think it would be very interesting to see those airborne concentrations, those 48-hour concentrations, because those could be informative, especially if we know what year those were happening. But people could have residual, could still have the pesticides in their homes.
Right, right. And Terry, did you want to speak to any of the, I know you looked at it, but if you're not prepared to talk about that now, that's okay too. Sure, yeah.
So we looked at, I think it was Wyatt 2003, and she has a table in that paper that presents the air concentrations. And the one particular table, 5 maybe,
I think it's table 5.
it splits out the air concentrations. If you look at the air concentrations from all the enrollees in these studies, the correlation between the cord blood and the maternal blood is not very good if it's all lumped together. The air concentration correlated with the blood levels. But if you split it out between more than a month before delivery and the month prior to delivery, then they were 48-hour samples, but they were taken at different times.
I was thinking it was 48 hours before delivery, but
yeah,
that
they're
would be crazy,
48-hour
of course. You
samples,
can't
but
do
they were
that.
taken at different time periods. So you've got samples, air samples that were taken more than a month before delivery, and then samples that were taken during the month of delivery. And the samples that were more than a month for delivery don't correlate with the blood levels. But the month prior to delivery, just like you're saying, it's a statistically significant correlation, but we're talking R.45, R.3, so it's a small R value, but they're statistically significant. So you'd have to argue a biological significance for those values. But then it sort of fell out, that month before. But then all of the analysis done after that, they lump everything back together, and they don't control for whether it was the month, more than a month or a month within a month of delivery. So the question would be, if you analyze that data in a time frame, whether you could dial down a little more on that effect, and I couldn't tell you that because they didn't do it.
Yeah, I think that there are ways one could pursue that. I think there's some very interesting thoughts there, but this is probably going too deep in the weeds at this point of the presentation.
Yeah,
And-
but that's
talk about that. Yeah.
And just one more note on that. I think, and Dr. Berry and I talking about this before today's presentation, what would have been probably the most powerful analysis they could have done is a pairwise analysis of each individual mother's air sampling and her plasma sample. And because they grouped all the data together and did that kind of analysis, it's really difficult to see if mothers with high plasma concentrations had high personal air sampling, or vice versa.
Yeah, I probably don't want to go into this in too much detail right now, but I actually think you provided some very interesting insights in how you looked at that, and I think that suggests some pathways to analyzing those data that we could talk about later.
So I would warn against making these correlations because we know that there's a lot of dietary intake as well. and you can get it through dermal absorption as well. So whether or not the air levels correlate with the plasma, I would actually doubt. So, but the other note I had on neurodevelopment, it's not just the early pregnancy, because part of the mechanisms might be neurite outgrowths, axonal and dendritic outgrowths, and that's especially at the end of pregnancy and early life. So we don't really know when the most vulnerable periods are. They could be very different, and they could extend into childhood definitely as well.
May I make one comment at this point? I went back looking at some of those human epi studies and went back to some of the methods that were published earlier. And I think I have some very serious concerns about whether those are even close to being on target. Those standard curves were generated out of water. There was a subsequent publication by the same group where the limits of detection were now 20-fold higher than what they had originally reported. So I think we should have some serious concerns about whether the data published by Wyatt, quantitative data, is really accurate.
Well, but any inaccuracy would not produce effects. It would actually hide effects.
I think based on what I see, the levels were actually probably quite a bit higher than what they reported.
If you do retain this table, I would suggest that you provide the 95% confidence intervals around the percentage of detectable chlorpyrifos. I actually think the only thing that really is relevant here is that they could actually detect any at all. And so, and since each of these proportions of case of participants in whom values were above the level of detection would have a distribution, and since these are relatively small samples, we should be provided with those data, if you might. It's pretty simple calculation.
So you'd ask that we do that analysis on these data or report if the authors did? Because if the authors did, I think we would have included it.
Yeah, you simply can do it. It's, 0.68 of 150 has a confidence interval that's easy to calculate. It's related to the square root of 160, and Dan can back me up on that.
So we'd be happy to. I just would make clear that if we are doing any analysis of independently authored data, that it doesn't appear like the authors published those data.
Put a footnote.
Just one more clarification question. So this chlorpyrifos level is singular chlorpyrifos, not attached to anything, not protein bound, not whatever. So could there be other types of chlorpyrifos in that plasma?
Well, you bring a good point, and I think this is what Dr. Buckpitt was alluding to, is that the samples taken were blood, then they were spun down, and then it actually was serum, not plasma. But there is a lot of matrix happening in a plasma sample that makes it a complex system. I realize that one of the papers that you sent us showed adducts, and none of these studies tested for chlorpyrifos adducts. However, when you have a standard curve made in water and your matrix samples collected from the subjects is plasma, there's a distinct difference in what your recovery will be and what the sample actually looks like. And to both Wyatt and Perez's benefit, they tried to receive serum and plasma samples from the American Red Cross and use those to spike and use to develop the standard curve. However, the samples they received from the American Red Cross blood donation program had enough residual pesticides in it, again from usually dietary sources, that they couldn't use them to develop their standards. So I thought you might find that interesting to note.
I do believe that they did denature those samples, though, so they precipitated proteins, which would knock anything off that was non-covalently bound.
Okay, moving on. So some summaries about the developmental, neurodevelopmental epidemiology in humans. Again, it's difficult to quantify associations between chlorpyrifos exposure and effects in humans because of all the issues that we have been discussing. So there's potential exposure to multiple OPs in the environment, and several of those OPs have similar urinary metabolites. And as Dr. Ritz mentioned, measurement of chlorpyrifos, and Dr. Hammond, or its metabolites at birth do not indicate what exposure might have occurred throughout pregnancy. And we do not know what the critical window of susceptibility is for neurodevelopmental effects with chlorpyrifos. And as others have mentioned, there's some inconsistencies with the analytical methods. But as stated in both our August 2017 and the latest draft risk assessment, we conclude that the epidemiological data alone are not sufficient to derive a point of departure for chlorpyrifos as a toxic air contaminant, but the data are compelling.
Can I ask you a question about that? But this is what EPA did in 2016, though, right? They used the epidemiological data to model a time-weighted average concentration and use that to determine a point of departure?
They did a complicated and very novel approach of dose reconstruction and back calculation. Eric, would you like to give a synopsis of what that methodology was?
Actually, in 2016, US EPA did adopt two different approach. First, I would like to talk about the April approach. So they assume a certain, they actually try to, using the Pbpk model, and then based on the exposure input, that prevalent at the time corresponded to the Columbium study, and then to calculate actually what the cord level looked like. Now, but the actual point of departure was, if I understood correctly, and Dr. Kuslukover can help me with that, they actually using the cord blood measure and then using the benchmark dose analysis to come up with a target. And then using the Pbpk model to see what would be the exposure in order to achieve that target in the cord blood. That's what they did in 2016 April. In 2016 November, they actually, at that time frame, they came up with another revision. So what they did is they, again, built on the spirit of the Columbia study, meaning actually the people that in the Columbia study experienced a certain type of exposure, and then using the US EPA standard residential protocol, the procedure to come up with exposure value, and then determine what would be the blood level look like, as a starting point, and then assume that blood level indeed associated with the developmental effect observed in the Columbia study. And then using the same Pbpk model, but this time it's completely reverse dosimetry, using the cord blood, and then to see what would be the exposure, end up at, not the cord blood, I'm sorry, the blood level. The blood level, what would be the exposure correspond to the blood level. So they started with, so let me clarify, they started with one exposure scenario called the crack and crevice, the hard surface. So they use that exposure, and then using the Pbpk model, and then to calculate what the blood level look like, and then use that as a target. And then based on this target, they're using the reverse dosimetry by the Pbpk model, and then to back calculate what would be the other exposure scenario, like say for instance, the dietary exposure. What kind of dietary exposure would achieve the same blood level that correspond to when they using the crack and crevice exposure scenario, come up with a blood concentration. I hope that clarify what they did. It's a little bit complex, but during that two USCB document, they did something very different. So is there any further clarification that I can?
Yeah, I just have a question. So, I mean, the November points of departure then were very low.
Yes. Because that actually, because of the way that it calculated, the November one actually, because they're using the 21-day time weight average. The concept actually built on the fact that based on the animal study, they couldn't actually determine the so-called window of susceptibility in terms of neurodevelopment. So I think that's the main reason, according to the USCPA 2016 document, that they use a time weight average over the 21-day period. And because if you taking the average of the 21-day, and also each day the exposure level is not assumed to be the same. Each day there's a 10% degradation, and the exposure is only for like 2 hours. So in the document it said shower, but what they really mean in terms of modeling is they cut off the exposure after 2 hours. So every day you have a peak, and then you drop another peak, but it's 10% less than the previous peak. And then you go on for 21 days, and then you average everything out, the 21 days. And because of that, the number turns out to be very low.
So I guess my question is, why didn't DPR use a similar approach?
When we dropped the document, I think at that time we using the we used the cholinesterase inhibition as an endpoint. I would say that it's something that I think it's fair to say that we will look into it, because it's a lot of assumption built in, for that analysis, because the 2016 November document it stated that it make the assumption that the crack and crevice exposure happened at that time, as associated with the developmental effect that they observed in the Columbia study. So the exposure really not the actual measurement, but it's rather an assumption. So I think that need to be carefully evaluated before we can move forward. So at this point, I think it is something that I think we need to look into a little bit more detail before we can make a decision whether this is something that we cannot be used for other purpose.
Yeah, I think that would be great if DPR could critically evaluate what EPA did and say, these are the good parts, these are the bad parts, and this is why we are or are not going to use this approach or something similar.
In the risk appraisal, there is a section where we try to provide that critical analysis. Of course, EPA are our colleagues, and we would be interested in knowing more about both the scientific methodology that they developed, as well as the assumptions going into this. However, they're at a point right now in their risk assessment where they put this out in November of 2016, so it's been just over a year. Then they entered a public comment period. Generally, what happens in the EPA risk assessment process for pesticides after that is then they have a scientific advisory panel meeting where that's their scientific review of what they've done. So they've finished we assume they've finished the review of the public comments. However, nothing has been published since then, and we haven't heard whether they're going to have a scientific advisory panel anytime soon. That's where these questions would definitely come in, and we have participated in those meetings either as simple audience members or actually on the panels before. And we would love to have that chance to have that discussion with them. But because their timeline for re-registration of Chlorpyrifos doesn't end until 2022, I would be actually surprised to see something come out from them before that time or closer to that time. I think that if they have an SAP, we'll certainly get notified about it so that we can put it on our calendars. without having more information about how they developed that methodology, it's hard for us to critique it. And if you go to the Federal Register, as we have, and you try to pull down the information about it, there's not as much there that we can figure out the entire method. So we're very lucky that we have Dr. Kwok, because he was able to go back through and piece together. And there's actually a figure, I think it's figure number 1 or close to that, at the very beginning of the risk assessment document you have, that shows this kind of back calculation and dose reconstruction and backwards dosimetry pathway. And that was, Eric deduced that from what we could figure out. Other than that, I wish we had more details about their methodology.
One of the problems with the back calculation type approach is that when you're starting out with these metabolites, you're not entirely sure that they were all originally from CPF. There are other chemicals out there that could have given rise to some of those metabolites. So it's very difficult to say whatever you back calculate was due to the Chlorpyrifos exposure. That's another issue.
And also, because there are personal air sampling data in the Wyatt study, it's a curiosity to me personally about trying to model those when you actually have air sampling data, empirical data. So putting that into the system and seeing what kind of number you get out would have been a very interesting comparison.
And I suppose that, going back to Beata's point too, is that these other routes of exposure besides airborne would be part of that. And then the composition of the science advisory panel's been changing in the last year. So that might also be affecting, I mean, apparently there were people on the panel who understood these things and suggested that US EPA do certain things, but now there's been some changes in the panel, correct? So that might be changing whether or not, how they look at this. And it might be that you might have to be a little more independent here in California for a while.
Just to the point of assumptions, I think all of these approaches make assumptions. The animal approaches make the assumption that you can model neurodevelopment from an animal in a human, and a human system that will be 70 years old, right? So I think what is most astounding is that all 3 human studies have been showing some effect on neurodevelopment and on a very complex type of neurodevelopment that we all are very proud of, right? The intelligence of humans. So, and how to measure that in mice, we haven't figured out yet. So I would give these human studies a little bit more credence on the level of the complexity of the system can only be measured in humans. There were blood lead, there was cord blood level measurements. Maybe they were very low, but maybe they're an underestimate of what there was. But obviously there's something going on in the human brain. And acetylcholine esterase inhibition is really only one of many, many, many types of modes of action that we can imagine. And we'll probably find out a lot more about what these agents can do. They are dirty agents. They're not just doing one thing, right?
Okay, moving on to hazard identification. Our risk assessment that you have in front of you addresses potential bystander effects arising from food and drinking water exposure, air and skin contact, incidental ingestion, and aggregate exposure from combined sources. We focus on 2 at-risk populations, females in childbearing years due to their potential pregnancy status, as well as children that are 1 to 2 years old because of their time spent outdoors and their potential for oral exposure due to mouthing objects and eating dirt.
Now I'm confused about bystander again. So this bystander is different than, so this bystander does not include the people, workers in the field, bystanders by the occupational and bystander.
This should include both residential bystanders and occupational bystanders.
both, okay.
What this doesn't include are agricultural handlers of the pesticide. Yes.
So just to be clear, it was implicit but not actually explicit in the report. These two groups of focus are because they're essentially the worst case scenarios, so that if you're modeling found levels that were protective for these groups, they should be protective for other groups. Is that correct? That was the rationale, right?
Yes.
These are the two. If anybody's going to be exposed to the highest aggregate levels with the most target organ toxicity, it would be these two groups. That's the stated the implicit rationale?
That is the implicit, that's the implicit
And
rationale.
it's not driven by we don't have good quantitative data for some of the other groups who, if we had the quantitative data, they might even be at more risk?
I'll let Dr. Koshlo go.
So there is one population subgroup that showed higher exposures through dietary sources, and those are infants, nursing, and non-nursing. However, nursing infants would not be exposed to the mouthing activities, foot to mouth and object to mouth. And so this is a significant part of the exposure, which makes the children want to the highest exposed population. In the aggregate exposure.
In the aggregate exposure, right?
If we protect this group, then we're likely to protect most people.
That's the...
I just want to be sure I understood because it's actually not stated as explicitly as that, I think, at least in my read.
We will add that in the next revision. Thank you.
We do have in the appraisal part, appraisal that infants have higher exposure to the dietary sources. So.
Okay. For the draft points of departure, for the revision that you have, the point of departure is defined as a dose that is not associated with adverse effects or that causes a low level of response. And points of departure, or PODs, are used as the starting point for determining risk. In our appraisal, we used a POD based on 10% red blood cell acetylcholinesterase inhibition. And we used human equivalent doses estimated by a I'll just say Pbpkpd model. Physiologically based, pharmacokinetic, pharmacodynamic model. And this model derived acute PODs for oral exposure and a derived 21-day or steady state POD for inhalation, dermal, and oral exposures. Briefly about the Pbpkpd model, it predicts a time course of chlorpyrifos metabolism in humans. It incorporates red blood cell acetylcholinesterase inhibition, reactivation, and regeneration after exposure to chlorpyrifos. The pharmacokinetic data were derived from human studies in both human liver microsomes and plasma that were used to represent metabolic variability across a broad range of ages, as well as inputs that attribute for life stages in infants, children, and adults. The model is able to do multi-route human exposure parameters, including oral, dermal, and inhalation. And it's undergone numerous scientific evaluations. The most recent update we received was in September of 2017. This is a list of the points of departure in our current risk assessment. And as I mentioned, the steady state inhalation for children 1 to 2 and females of childbearing age is the 21-day POD. So just to clarify that. The number you see here, 2370 micrograms per meter cubed, the air concentration, the point of departure, or if you calculate the reference dose by dividing by an total uncertainty factor of 100 to 23.7 micrograms per meter cubed, is the number that we talk about in the body of the document. However, in the risk appraisal, you will note that we have an additional discussion about this particular number. So I wanted to go into a little bit of detail about how we've changed slightly from the number as you see it here. First of all, Dow AgroSciences commented during the public comment period that the steady state 21-day inhalation point of departure, sorry, there's a misspelling there, for children aged 1 to 2 years old, presented in the US EPA 2014 risk assessment, would not achieve a 10% inhibition. In other words, it's not enough of a concentration in air to achieve that inhibition level. So in a separate analysis requested by our department, Dow used our default physiological parameters for children 1 to 2 years old to estimate the air concentration, which they came up with as 3,000 micrograms per meter cubed. And that was the number that they found resulted in 10% RBC acetylcholinesterase inhibition after 1 hour exposure for 21 days. Then we did our own independent analysis and to estimate a 1 hour per day, 21-day steady state POD value for inhalation. And we used the latest version, i.e. the September 2017 Pbpkpd model, and the model input parameters as were specified in the 2014 US EPA risk assessment, and came up with a resulting POD of 2850 micrograms per meter cubed. So altogether, you can see our previous number of 2370, and that's also the number that EPA used in 2014, and that's in 2 of our previous risk assessments. The number from Dow, which is 3,000, and the resulting number, 2850 micrograms per meter cubed. This is our new 21-day steady state point of departure that will be used by DPR for finalization of the Chlorpyrifos TAC document.
Just to clarify, and this will be something I think I will be coming back to and other panel members, but just to be absolutely clear, your PDPK modeling, which is based on human data, not another species, correct? And which gives you a point of departure for 10% acetylcholinesterase inhibition, which is the threshold at which one can be secure that it occurs, correct? So the PDPK model completely does not include any of the does not take into account any of the observations from animal studies that show neurodevelopmental adverse effects at levels below which we believe acetylcholinesterase is inhibited, and therefore it is as likely as not that some other pharmacodynamic mechanism is coming into play. Am I correct in that assumption of what you're assuming?
So there were multiple questions in there, so I'll try and tease them out and also probably rely on Dr. Kwok to answer some of them. So the current model, even with its most recent updates, uses the endpoint of acetylcholinesterase inhibition, not any neurodevelopmental effects.
Well, that's not exactly my question is one step removed from that.
Okay.
It relies on acetylcholinesterase at 10% and therefore presumes that any pharmacodynamic effect which occurs that is relevant is driven by detectable acetylcholinesterase inhibition.
I think that's a correct assumption. That's right, Eric?
Not so sure, because I think go back to the rationale of the PBPK model construction. Because, I mean, in general, you develop a model to answer the question that you're interested in. So in that, for this particular model, the model was designed to address the chlorinaceous inhibition. So the entire model actually built around that, the kinetic portion, and then how the kinetic couple with the pharmacodynamic, which the endpoint of concern is the chlorinaceous inhibition. That's all this, intended for. So anything outside that, I'm not sure that's.
Well, I'm quite sure it doesn't take into account anything outside of that. You're just confirming what would be logical based on what you're presenting. I just want to be clear that I haven't somehow missed the boat on that. That the entire precept of the PKPD model is acetylcholinesterase inhibition. In fact, you bring up a good point, which is actually there are aspects of the pharmacokinetic piece of this which could be, which also assume that what really matters is the metabolism insofar as it impacts the ability of this chemical to inhibit cholinesterase. So, for example, if it was a downstream metabolite that was actually causing a different problem, let's say acting as a cannabinoid receptor effect, and that wasn't from the parent acetylcholinesterase, that could be an issue. Similarly, if that effect was driven by the parent compound, but not by the oxime compound, which we know is a more potent inhibitor, then then you would sort of a pharmacokinetic model would have to take that into account too, in a different way than the standard pharmacokinetic piece of your PKPD model. Is that correct? I'm understanding all of that correctly.
I'm not sure that the focus of the model on chlorinaceous inhibition automatically discount the occurrence of, if I understood your comment correctly, discount any other effect. It really depends on actually how you use the model, because the model, in addition to the chlorinaceous inhibition, it can generate the Chlorpyrifos concentration in the blood. So if the blood turns out to be associated with other effects you're interested, you can also actually establish to see whether there's a correlation. So I think my answer to your question is that the intention is to look at the chlorinaceous inhibition, but it doesn't mean that the model can't discount anything, outside the chlorinaceous inhibition. You can always
on the pharmacokinetic part. Pharmacodynamic, yes. Without question, anything that's not acetylcholinesterase inhibition is not counted in this model.
Yes, that's
and
correct.
The other thing is a bit more of an open question. You'd have to delve pretty deeply into it to figure out the nuance of what's being weighted and how, right?
Yes.
Okay.
But this chart, what we're saying here is that an inhalation exposure at an average level of 2850 averaged over a 21-day period is the point of departure?
Well, rather, it should say that an exposure of 2850 micrograms per meter cubed for 1 hour a day for 21 days results in 10% acetylcholinesterase inhibition. Is that correct? Yes. So, and just again, to underline the point that the previous inhalation concentration for our 21-day POD of 2370 is now revised upwards slightly based on our reanalysis since the public comment period in September. On to the exposure assessment. Again, we looked at non-occupational bystanders, including residents. Short-term exposures, less than 24 hours from a single application. And there were two populations of concern again, females in childbearing age and children 1 to 2 years old. And we looked at indirect exposure associated with primary spray drift, including ground boom, orchard air blast, and aerial applications. And the routes of exposure that we assessed were dermal, oral, including non-dietary incidental ingestion by children, inhalation, exposures from food and drinking water, as well as aggregated exposures. The exposure assessment approach that we adopted is based on the US EPA spray drift methods published by Jeff Dawson in 2012, and that was to determine expected environmental concentrations, as well as the US EPA standard operating procedures for residential exposure assessment, or their SOP, from 2013 to do the exposure calculations. In addition, we used computer simulation modeling to estimate spray drift, both the horizontal deposition and 1-hour time-weighted average air concentrations. The spray drift models that were used include AgDrift version 2.0.5 and 2.1.1, which are empirical or curve fit models. And those were for deposition only, as well as for reflex application methods for both ground boom and air blast. And AgDiff's version 8.28, this is a well-vetted Lagrangian first principles model that follows the behavior of droplets after they're released from aircraft nozzles. The comparison of the AgDiff's output with measured field data have shown that the model tends to overestimate field measurements, so it likely is an overestimation of residential bystander exposure estimates, or it's a conservative approach. It also models deposition and air concentrations, and it can be used for application methods including fixed wing aircraft and rotary aircraft. We used reasonable worst case model inputs, including that the ground boom and orchard air blast worst case application methods were assumed as, and Terry, I apologize. We used aerial applications as the surrogate for ground boom and orchard air blast, correct? Correct. Sorry about that. But because of the data that we have, we looked at dormant apple applications, which is one of the highest rates of application, as well as high boom, ground boom application. And for the aerial application, the reasonable worst case, it was based on reasonable worst case agricultural aircraft types in California and based on a DPR enforcement county survey. So these are kind of, the modeling is based on actual use cases in California. And we also use real world meteorological conditions for the San Joaquin Valley, which were chosen to produce the highest model downwind application or deposition. So putting together the risk assessment and the PODs, we were calculating risks as margins of exposure, which are the ratio of the point of departure to the estimated human exposure level. So the POD we mentioned as 2850 micrograms per meter cubed, and the estimated human exposure derived from the modeling. A target margin of exposure of 100 is generally considered protective. And for this particular risk assessment, that target uncertainty factor allows for the following: 1 for an interspecies sensitivity, 10 for intraspecies variability, and 10 for potential neurodevelopmental effects. The margins of exposures were calculated from route-specific points of departure that were mentioned earlier, and we also used aggregate or combined MOEs calculated through exposure to skin, skin contact, mouthing activities, breathing, eating, and drinking. In all of these calculations, we determined that 2 exposure scenarios had we estimate to have no health risks, or that were above the margin of exposure of 100, and no risk to our at-risk populations of children and women in childbearing age. And those include dietary exposure, only, that means residue in food and drinking water, or exposure from dermal exposures resulting from spray drift. However, our calculations came up with results showing that exposure scenarios, several exposure scenarios might have potential health risks or that have margins of exposures below 100. And those include hand-to-mouth exposure in children, meaning the incidental hand-to-mouth activity, eating dirt, mouthing objects, etc. Inhalation exposure to children and females of childbearing age, and various aggregate exposures from combined media, including food, drinking water, and deposition from spray drift. And it's important to note that the exposure to aerosols in the air near application sites was the main driver when the aggregate MOEs were less than 100. That's why this is being considered as a toxic air contaminant. The key conclusions from our risk assessment are as follows: that the database on chlorpyrifos inhibition of acetylcholinesterase is extensive. It also supports the establishment of dose response that can be recreated at specific dose levels in animals. At this time, it is not possible to determine a quantitative dose response or dose effect relationship based on any human endpoint. Therefore, these studies cannot be used as the scientific basis for our regulatory target. The lack of dose response or a clear mechanism of action does not negate the results showing potential associations between in utero human exposure and human development, and new data will be evaluated as they become available. This bullet above is the basis for why we chose a 10-fold uncertainty factor for neurodevelopmental toxicity. And new animal data identifying developmental neurotoxicity at doses lower than acetylcholinesterase were added in the recent version. And new data that we didn't include in the version are also being analyzed right now. We will continue to consider the suitability of deriving a point of departure specific to developmental neurotoxicity in the final toxic air contaminant evaluation document. Those are our key conclusions.
Thank you.
Can you go back to that slide for a second? When you write at the bullet number 3, at this time it is not possible to determine a quantitative dose response or dose effect relationship based on any human endpoint. Therefore, these studies cannot. Do you mean any human endpoint of neurodevelopmental toxicity? I mean, the human endpoint that you're using is acetylcholinesterase inhibition. I mean, is that?
You're correct. I should have clarified that human neurodevelopmental endpoint.
Uh-huh. And the last one, which is we're also not using any experimental animal neurodevelopmental endpoint either at this time, but we might. Is that what you're saying?
That's what we're saying.
But wouldn't that completely change everything that you've done if you do that?
As our meeting with Stan Glantz pointed out, it would require a complete revision of this document.
Uh-huh. So.
Oh, well, wait. Okay. I just want to say first for the record, all the questions I was going to ask about got asked by somebody else, so that was great. But what I was suggesting was that one would not want to have to go do a complete revision of the whole document. Okay. But I do think that because I think that the neurodevelopmental endpoints are very important. And what I was, talking about on the phone call we had was whether that could be taken into account by adjusting the uncertainty factors or something short of going back and completely revising the document. So I just want to.
And we actually have a table. We developed a table to try and answer your question, to show what our current POD is, what it would be if we use the animal DNT study and compared to EPA's 2016 number. And we'd be happy to bring that up now for discussion or later after OEA has a chance to go over their findings.
Okay, well, it's really nice that you did that. I'll leave it up to the chair to determine when you present that, but I'm looking forward to seeing it.
Okay, so
So is Kathy.
on that note, what I'd like to do is, while OEA is setting up to discuss their findings on the document, that we take a 10-minute recess for getting everything turned over and give people a chance to deal with biological necessities if necessary.
Is that sufficient for our stenographer? Is 10 minutes sufficient? Okay, thank you.
Thank you. Okay, I'd like to call this meeting back to order. If everybody will take their seats, please. All right, the next thing on our agenda is a presentation on Oia findings on the draft TAC document, and that'll be presented by David Ting and Lori Lim, and I'll turn it over to you.
Good afternoon, members of the Scientific Review Panel. I am David Ting, Chief of Pesticide and Environmental Toxicology Branch at the Office of Environmental Health Hazard Assessment, or OIHA. Sitting to my right is Dr. Lori Lim, Chief of Pesticide and Food Toxicology Section. We are here to discuss with you OIHA findings on DPR's draft TAC document for Chlorpyrifos. Also in the room are Dr. Heather Bolstad, Dr. James Nakashima over here, and Dr. Rima Woods, and Dr. Weber Laribi. They are part of the team who carry out the review. As you have heard from the previous presentation, DPR, okay. As you have heard from the previous presentation, DPR has developed a draft risk assessment on Chlorpyrifos, and it will be used in considering whether to identify the chemical as a toxic air contaminant. OIHA is required by law to prepare findings on this risk assessment. Our findings have been submitted to the panel and DPR, and they are also posted at OIHA and DPR websites, as indicated on this slide. Overall, we agree with the approach of using margin of exposure to quantify the risk of residential bystanders exposed to Chlorpyrifos. Our primary recommendations cover evaluation of additional toxicity endpoints and re-examining how to address the uncertainties in the assessment. We also agree with the determination that bystanders can be exposed to air concentrations of Chlorpyrifos that exceed the health protective level. Our findings support the identification of Chlorpyrifos as a toxic air contaminant. With that, I'm going to turn the microphone over to Dr. Lim. She is going to describe in detail some of our more important findings.
Hi, I'm Lori Lim. Is that better? Okay. Hi, my name is Lori Lim. I will be presenting the findings directly related to the determination of risk for bystanders' exposure to Chlorpyrifos. They are toxicity endpoints, points of departure, exposure assessment, and uncertainty factors. Next. So my presentation will start with finding number 8, which is using the RBC acetylcholinesterase or ACHE inhibition
as the critical toxicity endpoint for chlorpyrifos. RBC ACHE activity is a sensitive marker of systemic ACHE inhibition. It is often used as a surrogate for ACHE's activity in the nervous system because it is easy to take blood samples, and the enzyme is essentially the same forms in both tissues. Toxicity studies show that with chlorpyrifos, RBC ACHE is often more sensitive than brain ACHE. The data in Table 1 illustrates this point. The experiment was conducted with two groups of young rats postnatal day 11 and adult female rats given chlorpyrifos by gavage. At 0.5 milligram per kilogram per day, there was no significant inhibition for either RBC or brain ACHE, indicated by the low values under each of the three groups. At 2 milligram per kilogram, more inhibition was measured for RBC, indicated as minus 36%, 31%, than for brain, which is 2% and minus 7% for the pups. In addition, more inhibition was detected for the pups' RBC inhibition compared to the adult RBC. So we have 36 and 31 for RBC at 2 milligram per kilogram versus 19% for adult female. This is one of the data sets to illustrate this point. The next finding is on developmental neurotoxicity, or DNT, as a potential critical endpoint for risk assessment. There are numerous animal and human studies showing exposure to chlorpyrifos is associated with DNT. And I'm saying that DNT is a simple developmental neurotoxicity, but it encompasses many effects. In some rat and mouse studies, chlorpyrifos exposure during gestation and postnatal periods was found to cause DNT effects on cognition, anxiety, social behavior, and motor activity. In some studies, these DNT effects were observed at doses that show minimal or no brain cholinesterase inhibition. This point was talked about with Dr. Duto's presentation. This mainly referred to a study by Carr et al. In 2017, in which the neurobehavioral effects were observed at 0.5 milligram per kilogram per day, but brain acetylcholinesterase inhibition were 4% decrease at 0.5, 4% at 0.75, and 19% at 1 milligram per kilogram per day. Oia's view of the DPR observations from a registry study and those in recent published literature are presented in Table 2 on the next slide. These recent animal studies were conducted with low doses, which can provide the basis for improved dose-response relationship analysis compared to the earlier studies, which are mainly conducted with either a single dose or high doses. In this table, the first study, Hoberman 1998, is the FIFRA guideline study, and the rest are published reports. GD is gestational day, LD is lactational day, and PND is postnatal day. In the Hoberman study, the most sensitive endpoint was reduced parietal cortex thickness in the pup measured on day 66 days old. Note that the effective dose is set at 1 milligram per kilogram. And there are no data for the lowest dose group of 0.3 milligram per kilogram per day. The key points about the published studies are all were conducted by the oral route, and a range of doses were tested. The pups were exposed to chlorpyrifos during gestation and or postnatal period, but were tested much later after exposure. The lowest effective dose was at the same level of 0.0 milligram per kilogram per day from 4 report. Effects at the low doses were anxiety, motor activity, and spatial learning. Next. There are human studies to support the consideration of DNT as a critical endpoint. There are epidemiological studies looking at organophosphate exposure and toxicity. 3 major prospective post-cohort studies have shown associations between exposure to organophosphates, including chlorpyrifos, during pregnancy and adverse neurodevelopmental outcome. The exposure levels were generally measured as metabolized in the urine. The Columbia study had the most comprehensive measurement in that it measured chlorpyrifos as well as TCPY, the specific metabolite of chlorpyrifos. Chlorpyrifos was measured in the maternal blood within 1 day of post-delivery and fetal cord blood at delivery, as well as TCPY in the urine of the maternal and fetus, as well as the urine and meconium. However, it is difficult to use these exposure data to establish direct dose-response relationship using approaches for laboratory animal studies. One problem is when the chemical level is measured for a single time point, while the associated effects are measured much later. Additional evidence of DNT for chlorpyrifos comes from zebrafish studies and in vitro model for DNT. Zebrafish requires a certain level of ACHE for normal development. When zebrafish is exposed to chlorpyrifos, it showed abnormal behavior and inhibition of ACHE activity. There are many possible modes of action for chlorpyrifos-induced DNT effects, but there is no definitive conclusion. This can explain the variety of DNT effects observed in human and animal studies. Finding 13 is a summary of findings on critical endpoints. For all exposure scenarios evaluated in the draft TAC document, the critical toxicity endpoint used was 10% inhibition of RBC acetylcholinesterase activity. While ACHE inhibition is a sensitive toxicity endpoint, other endpoints such as DNT and respiratory toxicity effects may be more sensitive as well as toxicologically more relevant. Next. The next topic is point of departure for the critical toxicity endpoint. The point of departure is the starting point of the low dose extrapolation and is used to determine the health risk, calculated as the marginal exposure associated with a certain exposure level. Generally, the duration of the point of departure matches that for human exposure duration. In the draft TAC risk assessment, the acute single-day inhalation, dermal, and incidental oral exposures were evaluated using steady-state point of departure for RBC ACHE inhibition. The use of the steady-state point of departure is said to compensate for background exposure to chlorpyrifos and cumulative RBC ACHE's inhibition. Oia finds that this is a conservative approach, but notes that it may add uncertainty to the risk estimate for this point. That is, the actual exposure is adjusted by the toxicity or point departure rather than by a better estimation of the exposure level. Another point from finding 14 is that since the point departure was derived from a Pbpk model, it would be helpful to explain the factors driving the values. For the inhalation exposure, the point of departure expressed as air concentration is about 2.5-fold lower for children than females. However, the point of departure in terms of dose, milligram per kilogram per day, adjusting for respiration rate but not for the 1 hour per day exposure, so for the two age groups so that they're very similar. On the other hand, the dermal point departure for children is more than 5-fold higher than that for female group. Finding 15 is related to the last point on the steady state of point departure, the uncertainty for the values. In the inhalation component of the model, Chlorpyrifos is modeled as dry particles with relatively small sizes. In contrast, the bystander's inhalation exposure to Chlorpyrifos is a spray drop cloud comprised of liquid aerosol droplets of varying sizes. The pharmacokinetics of Chlorpyrifos of these two forms could be different at different deposition sites. In addition, the steady-state inhalation component model has not been validated. There are no subchronic inhalation animal or human toxicity data suitable for this purpose. The only human inhalation exposure toxicity was a combined acute dermal and inhalation study by Vaccaro et al. This study will be discussed in finding 22. Finding 16 is on exposure assessment. The acute exposure is for 1 to 1.5 hours per day. The TAC document estimated exposure for individual routes and all routes for spray drift, shown in the first row. In addition, it included an spray drift and dietary aggregate exposure in the second row of the table, but only for children 1 to 2 years old, but not for female adults. While children often have the higher intake on a body weight basis and slightly a more sensitive group, the risk of these two groups can't be compared based on exposure levels alone. The risk for the female group needs to be determined given that the point of departure for this group are different than those for children. Finding 17 is also on exposure assessment on the use of surrogate air concentration. We agree that the use of the surrogate aerosol concentration is appropriate. The concentrations are likely conservative estimates for the ground boom and air blast applications. It is the best available method to estimate the air concentration for these applications. They are similar to air monitoring data by the California Air Resources Board for air blast application. However, they could be underestimates for a specific scenario when there is little or no foliage, resulting in more spray drift than predicted. Finding 19 is on exposure. In this case, the need to consider Chlorpyrifos vapor exposure. The concern is for residential bystanders who also live close by. There's been several questions about what's the definition of bystander. And the way we are interpreting it, a residential bystander is somebody who can be walking by the field as well as living near the area. They can have aggregate exposure to both vapor and aerosol. Release of Chlorpyrifos as vapor from deposits is possible in areas where the summer temperature is high. The relationship between temperature and vapor pressure is shown in this graph on the slide, where increasing the temperature from about 70 degrees to about 100 degrees causes a 14-fold change. And this exposure to Chlorpyrifos vapor may last for many hours compared to the 1 hour exposure duration to the aerosol. Thus, the effect of temperature on Chlorpyrifos is an important factor to consider. In addition, we want to note that the US EPA added vapor exposure estimate in the 2014 revised health risk assessment. The remaining slides of this presentation are on uncertainty factors, starting with finding 22 on interspecies uncertainty factor. This uncertainty factor is an extrapolation factor used when data from animal study are used to extrapolate to human response. The full factor is tenfold and is reduced to 1 when adequate human data are used. Oia recommends that interspecies uncertainty factor threefold because there are uncertainties in the Pbpk model which simulated the pharmacokinetic and pharmacodynamic of chlorpyrifos in human. The model is not equivalent to a well-conducted human study because some of the model parameters were derived from animal studies. There were limited human data for model validation, as will be shown in the next slide. Furthermore, the model has not been adequately validated for human steady state exposure for any route. The table in this slide is a summary of the human studies presented in our finding. It shows the acute human studies used to validate the acute and steady state outputs of the Pbpkpd model. The data from the first study were also used to derive model parameters. They're the Nolan et al. Studies for the oral and dermal route, the Carcinogenic et al. Study for the oral route, and the Vaccaro study for dermal and inhalation combined. The key points of these studies are there are few subjects and they are all adults. 3 of the 4 studies use single dose, one-time acute exposure. RBC-ACHE's inhibition, the critical endpoint basis for the point rupture, was observed in only 1 subject and in 1 study, and that's the one by Carcinogenic et al. It should be noted in the Vaccaro study, a slow-release formulation was used. This formulation shows lower peak air concentration by about 4-fold and much lower toxicity than non-encapsulated chlorpyrifos. In this study, the subjects were exposed to chlorpyrifos by dermal and inhalation routes. So the subject wore suits in which they roll around the floor to simulate kids' activities. And then the materials cut up and measured for dermal absorption. More importantly, RBC acetylcholinesterase activity was not measured. Finding 23 is on intraspecies uncertainty factor, which is used to account for differences in response to a chemical between humans or inter-individual variability in the human population. An intraspecies uncertainty factor 30 is needed to fully account for the potential variability in both pharmacokinetic and pharmacodynamics in the human population response to chlorpyrifos. The Pbpkpd model did not fully account for the physiological, anatomical, and biochemical changes during pregnancy and among different age groups. For pharmacokinetic considerations, sensitive parameters related to metabolic clearance of chlorpyrifos and its oxone were based on in vitro data from a small number of plasma and liver postmortem tissues as shown in this table. Note on this table that there are no more than 10 samples per age group, and for the age group of children 1 to 2 years old, there were only 1 plasma sample and 5 liver samples. In addition to pharmacokinetics, there's a need to account for the variability to the pharmacodynamic aspect of the RBC-ACHE's inhibition. The reported coefficient of variations for the parameters describing the inhibition are relatively small or small. For example, the inhibition rate was derived from all male adults. It is unclear how representative the mean and CV values are for the general population. It is known that RBC-ACHE activity varies with age, pregnancy, and even between healthy adults. Another consideration is whether the intraspecies uncertainty factor derived from RBC-ACHE inhibition would be applicable for DNT. Many factors can influence an individual's susceptibility to developmental neurotoxicants, potentially resulting in a large inter-individual variability. The final finding is finding 24, an additional uncertainty factor. This factor is applied because the DNT may be a more sensitive endpoint compared to RBC-ACHE inhibition. This factor is part of the Food Quality Protection Act on the evaluation of pesticide tolerance in food. The Act requires the U.S. EPA to apply this factor when there's potential pre- and postnatal toxicity and incomplete exposure and toxicity data for infants and children. In the draft TAC risk assessment, the tenfold factor was applied because the DNT data were considered inadequate to use to form the basis for a point of departure determination. In our view, the use of this factor in a surrogate endpoint of RBC ACG's inhibition adds uncertainty to the risk characterization. Thus, in light of the more recent published report, as well as the body of evidence of human epi studies, we recommend a thorough evaluation of these DNT studies to see if a point of departure for DNT can be directly determined. This ends my presentation. Any questions?
Thank you. So let's open this up for some brief questions by the panel.
Dr. Lim, I had a question on your, I think, fourth slide, the finding 8. Are there any data out there in animals? You have inhibition here in pups and adult females. From 2 milligrams per kilogram, that's a single dose, right? Are there any data out there for multiple doses and a dose response?
This is the dose represented, what the group has is 2. And then in this same experiment, another group had 0.5.
Right.
Yeah.
But are there any studies where they give 0.5 per day for several days?
Yes, this is a multiple. This is a single day experiment, but there are multiple day experiment. It's quite detailed described in the TAC document with multiple doses.
Okay.
Okay, thank you. I have a question actually related to the Oia Findings document. Can I ask you about that? So on point 25, the last page, page 14, okay, so the you say that the target MOE would be 1,000 based, well, actually that's based on the Oia analysis, right? The uncertainty factors you were just talking about, factor of 3, a factor of 10, a factor of 30, gives you a factor of 1,000. 25, sorry, the Oia finding document?
It's the finding memo itself.
It's the December 2017 document. Oh, that's not it. That's DPR's response to OEA.
You have us a lot of documents.
We're trying to make it short.
I appreciated that. Did Alan? Okay. Okay, so based on the uncertainty factors for DPR, they came up with a total uncertainty factor of 100. Oia is saying total uncertainty factor of 1,000. But then in that last paragraph, the first sentence, consideration of Oia's above findings on the uncertainty factors could result in a higher TAC target MOE of at least 10,000. Can you explain the 10,000?
It is very confusing because we have health risk assessment, okay, which we have the standard, uncertainty factors. But under the Toxic Air Contaminant Act, the criteria for it to be listed is tenfold lower. So it flips the equation to meaning that you require a higher margin of exposure.
Sorry, so this is an additional factor of 10?
Yes.
Over the total uncertainty factor?
Yes. That's why we specify that as a TAC target versus a regular target. Because the MOE is the point departure divided by the exposure.
Right.
So normally with the DPR document, you want that to be a 100-fold difference. That's the 100. But under TAC, you want that difference to be 1,000. So effectively, you're dividing the point departure by 1,000 to get the reference.
Right, but I had the same question, so I could see that, but then I said, are you saying that the margin of exposure should be 10,000? Because that's what it seemed like you were saying at the end of that sentence, so is
If
that
you
what you guys
apply
are
this, yeah, if you apply the math and what the TAC criteria is, then it would be 10,000 from our 1,000, yes.
I think maybe it might be clearer if you laid out each of, there's so many places the uncertainty factors are coming in, interspecies, different ages, all of these. If you were actually to lay them out, there's this factor, there's this factor. At one point there was a 30 factor, so I'm surprised we don't end up with a 3 in the front of 3 times 10 to whatever it is. But if you were to just kind of lay each of the factors out, it might clarify for everybody.
That's a good suggestion. Just to get back to your question, the first paragraph of our finding number 25 explain why this additional 10 is specific to the TAC program.
And that's simply to be listed as a TAC.
Yeah, that's the criterion for that program.
Right. That's not to say that the reference concentration should have that 10-fold factor. This is purely for the listing criteria.
I'm now totally confused. Maybe the thing to do, because this is clearly going to be one of the big issues in this, maybe over lunch you and DPR could make a slide with each of the different elements that contribute to this, and you could have the OEI number and the DPR number next to each other, and then we could just see why do you think different things?
Yeah, I think.
Because I mean, I could, yeah. I mean, I still don't understand where that. I mean, my understanding of the way the margin of exposure worked is you have the point of departure, which is kind of like what OHIA likes to talk as the reference exposure level. And then when OEA and ARB are doing it, they divide that to say what's the acceptable level, and what DPR does is they flip the fraction, and they ask like how much below that are you, but I don't see how that should make a difference, changing 1,000 to 10,000.
But I think if you take findings,
it seems like they ought to be just invert. They should just be inverses of each other, just because it's a definitional thing. I don't see why it should make such a gigantic difference in the number.
But I think if you aggregate the recommendations from finding 22, where you're going to increase the interspecies uncertainty factor to 3, and then finding 23, where you're going to have an intraspecies uncertainty factor of 30. That gives you a 90, then the other 10 gives you 900, and then the original 100, you get up to 9,000. And so there may be a little bit more play in there as well.
Can I ask?
We'll try to make
still
that
a pretty big difference from what DPR was saying, so I think.
We'll try to make that table as you have instructed.
Okay, and because I think that's going to. I mean, there's a couple of key issues that have come out of all this, and that's one of them. I mean, to me, the two key questions that I had going into this were the neurotoxicity endpoint versus the acetylcholinesterase endpoint, and the other one is what's the appropriate uncertainty factor. I think after reading through all of this, and then there's the questions about the exposure model, but in terms of the biology, those are the really two key questions that I think come out of this. So I think in the discussion after lunch, the more we can get those differences ventilated and explained, I think that'll really help move things forward. Because I still don't see where you get the 10,000, but we don't have, we can do that after lunch.
So that's assuming there is a lunch. I
Kleinman
want to
said
just
there would be
clarify
lunch.
with you in the same way that I tried to clarify with DPR some things, so see if I understand and can grasp the differences and the similarities. So in terms of the modeling that was used for pharmacokinetic, pharmacodynamic relationships, your point is that the some of the input parameters in that model are in fact derived from animal studies, not from human studies, and therefore a value of 1 is not sufficiently conservative because you're not using a human model. Do I understand your point correctly on that?
Yes. Using uncertainty factor of 1 implies that the prediction is right on. There's very little error. And in our review, this model has certain limitations.
Right,
They did
but the limitations are not solely that it was validated with a small number of human observations, but also that some of the parameters were derived from animal studies, not from...
we listed a couple of things,
Yeah, okay.
including the different in nature of the particles used in the model validation and the real situation, the difference in terms of not all parameters are derived from human samples,
Yes,
as
okay.
well as small number of samples.
Okay. So, all right, so that's essentially what I understood you correctly. Okay. None of that critique addresses the pharmacodynamics being based on acetylcholinesterase inhibition. You seem to be accepting that as an appropriate pharmacodynamic component to a PKPD model, or am I missing something?
Actually, we try to separate PK from PD.
But you said to increase the uncertainty factor by 3 because of the PK, not because of the PD. Otherwise, you would have come up with 10 instead of 3.
We accept the model as a model for RBC cholinesterase inhibition.
Why would you accept that?
Because that's what it was intended for.
But
And
that's what's
so-
being applied here.
Right. And I think if we would say we cannot use this model for DNT, you would need a different model. So we wouldn't apply an interspecies for that reason.
So therefore, what are you suggesting for the modeling of risk here? Are you just throwing up your hands and saying there is no way to do it?
No.
Because all of your suggestions about adding additional uncertainty factors still presume that they're using modeling of acetylcholinesterase to model the endpoint that you say they should model, which is neurodevelopmental toxicity. So it seems to be a contradiction of some sort, an internal inconsistency in your argument.
You are right in saying that we explore both options. In using, keeping the RBC cholinesterase inhibition as the point of departure, we suggest a bigger uncertainty factor. However, we also realize the cumulative uncertainty factor would be relatively large, so we recommend to explore the possibility of using DNT directly.
Of using what? I'm sorry?
Developmental neurotoxicity as an endpoint directly.
But that's what DPR suggests by adding a factor of 10, an uncertainty. That's where their uncertainty factor of 10 comes from, isn't it?
Yes, as an additional uncertainty factor. We're actually looking to the panel to give us some input on, as part of your charge question, to ways of looking at the EPI study.
But, okay, so I guess what you're saying is you can't answer my question.
Now, we understand the uncertainties. We understand and what USEPA did in their two assessments in 2016. And it's a really difficult question. You really need to have the raw data from the EPI studies. And I think we have greater confidence in the human studies at this point, but if there are ways to look at the EPI studies.
You mean the non-human studies?
Yeah, then we certainly will explore that.
Uh-huh. So you don't mind it. Can I keep going with this line of questioning? Okay. So assuming that you're saying, hypothetically, were you to stay with acetylcholinesterase inhibition as the endpoint, even though we don't think that necessarily should be the endpoint, but you could maybe address that by adding a factor of 10 for the neurotoxicity. You've then suggested that for intraspecies, instead of using 10, you use 30, plus 10, another 10 for neurodevelopmental special things. And the reason why you're saying use 30 is because of childhood vulnerability, right? In terms of cholinesterase sensitivity of effect?
Yes, it's
or
all,
a vulnerability to effect?
It's all about the RBC, acetylcholinesterase inhibition.
Right.
Yes.
Now, doesn't using a intraspecies factor of 10, which is a fairly, that's the high end of what's typically used, especially if you've got another factor of 10 for the specific subset of neurodevelopmental toxicity. I mean, usually 10 would be about as high as you go for intraspecies, which would generally take into account childhood vulnerability or special subgroup vulnerability. So is there a specific rationale why OHIA suggests 30 instead of 10?
So some of our reasons are explained actually in finding number 23. We separate them into pharmacokinetic and pharmacodynamic considerations. But to answer your question directly, a number of considerations. First of all, RBC acetylcholinesterase is being used as a surrogate. So it is a little bit unusual. And second, if we do not have specific reason, we may choose to use a smaller factor. But in this particular case, we have pretty strong data to indicate there is developmental neurotox. But
that's where the factor of 10 comes from, right?
But we see challenges in doing quantitative dose response. That's why we think 30 is appropriate.
So where I'm going with all of this badgering of you is, isn't the logical conclusion of what you're saying that the interspecies factor, which you're suggesting should be 3 rather than 1, because of your uncertainty in the toxicokinetic portion of the PKPD model, pharmacokinetic part of it? I think that's what I heard you saying, right? It's got to be either one half or the other half that you don't believe the data are certain about. Because otherwise you'd say it's a value of 10, not 3.
Actually, that number, our consideration is the model uncertainty. Is another way to say the same thing.
Which part of the model?
The conceptual model of the PBPK, how it's implemented, how it's parameterized.
Why wouldn't you have said it's 10 and not 3, given everything you've said about it, and given that it's looking at the wrong endpoint or the wrong mediator?
I think it's the state-of-the-art PBPK model, and a lot of effort has gone into it. So it may not be perfect, but we would like to give it some credit in try to.
I think 10 is giving it a lot of credit based on everything you've said.
anything you want?
Okay. Well,
You have to introduce yourself.
That is now, yeah. Okay, my name's Andrew Salmon. I'm a senior toxicologist with Oia, and some of you may recognize me having worked on the air program for a year or two in the past. I just wanted to add what I see as a bit of a clarification about what these various uncertainty factors are. And the way we wrote this up for the Hotspots program, which is the program that has the defined guidelines where this is all laid out, was that we talked about the uncertainty factors, firstly for interspecies, and conventionally that overall factor has been associated with a value of 10. And within that factor of 10, there are two components, one of which is a pharmacokinetic uncertainty with a value of the square root of 10, which is conventionally reduced to 3. And the other is a toxicodynamic uncertainty that is conventionally associated with a value of root 10 as well. So that's the I think in this case what we're saying is that the uncertainty which David was describing as the factor of 3 is the, or square root of 10, that is the toxicokinetic component of that uncertainty factor. And there's also a factor of 10 going into the toxicodynamic component because of the uncertainty of inter-individual variability. And that's a excuse me, that's a component of the intra-species, so it's a different uncertainty factor. So we've got a we've got residual uncertainty in both elements here. Exactly how they've assigned these various values is a little bit of a decision that is laid out differently here. But I think the point I'm trying to make is that it fits the standard model that we've got the interspecies uncertainty covered, and that's one factor. And then we've got the intraspecies where we've got toxicokinetics and toxicodynamics. Intra. Well, there's
both.
Yeah, there's uncertainty to be accounted for in both areas, both in the interspecies extrapolation and the intraspecies.
Right.
So that's why you've got 3 or root 10, and then you've got then you've got another one.
And I just don't remember you coming up in your brief tenure with the organization. I don't actually remember a situation, and I could be misremembering, where we had a value for 3 for interspecies and a value of 30 for intraspecies and a value of 10 for further uncertainty. Yeah. I do remember a lot of examples of 10, 10, and 10.
Yeah.
And
I think this case is very special because we're talking about a chemical which is starting with a surrogate endpoint as a basis of point of departure.
I think that's right, absolutely.
In the program, you start with the most sensitive endpoint, and then you add the uncertainty factors to that. So that's why our main recommendation is let's get the point of departure right with the best endpoint, the most relevant endpoint, and then we'll work on the uncertainty factors.
Yeah, I mean, I think this is, as you say, is a special case, and in terms of modeling the cholinesterase endpoint, we probably, in spite of our reservations about the PK modeling and all that sort of thing, we actually have a rather better coverage of that specific topic than we typically did with some things in the AIR program. But the big extra issue in this case is the fact that we're essentially relying on, for this assessment, we're relying on a substitute endpoint, and the AIR program would, had the AIR program been using the hotspots model for laying this all out, we'd have probably put that in as a data uncertainty factor. We'd have called it a separate factor of 10 because of the uncertainty about extrapolating from a surrogate endpoint to the one that we were most concerned about. So I think that's where that extra one would have been put in.
And that's aside from the factor of 10 for neurodevelopmental?
No, that's where that comes in.
Right. So algebraically, I'm not actually that far apart in this view. That is to say, I think it should be 10, 10, and 10, and you're saying it should be 3, 30, and 10.
Yeah, there's room for debate about that.
I think by precedent and by clarity, my own view is, were you to recommend 10, 10, and 10, it would be more elegant.
Well, I think,
and it would all. Speaking as one of the people who helped develop those rules of thumb back somewhere in the Pliocene age, I think it would be a lot clearer. I mean, you're going to get the same answer.
Yeah.
But I think it would be a lot clearer, and you would also be able to cite, all of the work we did in developing those uncertainty factors and defining them before. Yeah. You end up in this. I still don't understand where the 10,000 came from, but I'll leave that for later.
Well, let me revise my request, the initial request, to get a table that lays out each uncertainty factor. And I like the idea of doing it from the DPR and from OIHA's point of view. But let's also do it separately for the two outcomes, the acetylcholinesterase inhibition and for the neurotox development. So those are two different outcomes, and they each have different uncertainties in them. And I think that's what's contributing to this kind of confusion.
Yeah, I think that's a really good suggestion.
So I mean, I think, and the fact you can't just sit, nobody's been able to sit there and make that table while we've been talking for 20 minutes on this says something about how complex it is to you two. I just say that.
We're making it right now.
So make it, and as I say, but I would suggest now that but we just need clarity. And I don't think doing it orally, I think, is not the way to do it, but make the table and have it for each of the outcomes, as well as if the different agencies have used different factors, just so we can see.
And I put it on the screen.
And then I have another question. In other iterations of my life in different places, the intra-species variation, so for instance among humans, has been a factor of 10 in workplace values. So in other words, that's all adults, and we're saying there's intra-species variation among adults, human beings, that relates to metabolic pathways and different aspects of that. So if that's true, I don't think 10 is appropriate if you're also going to children. And certainly fetal development, I would imagine, is something more. So I guess I don't know if now I can't remember because I was part of that too, but I forget the hot spots thing. But I think that there's an adult 10 and then something else for children.
The way the hotspots default works is that the overall value for the intra-species uncertainty factor, the UFH, is 30, which consists by default of a square root of 10 for the that's it. There's an uncertainty factor of 30 overall. Let's not worry about that. Overall 30 default for the whole factor, which includes both the toxicokinetic
Right.
and dynamic.
So yeah, I think for me, I'm just a little visual. It'd be helpful to just see it up there,
I think
and if we could, do that. And then I have one other question that's along this line, and that is are we thinking at some level that the inhibition of acetylcholinesterase is a step towards the neurodevelopment problem? Or is it just something that is, this is a chemical that has this effect and has this effect, and they're unrelated to each other?
As we pointed out, there could be many mode of actions for developmental neurotox. I think US EPA counted as many as 5 or 6. But there's no definitive answer to that question. And inhibition of acetylcholinesterase may or may not be one of those. I don't know the answer.
Right, and then let me just ask the question. I won't say what I think. Do we know whether it's a direct action or a metabolite that's responsible? I didn't think
We don't
so,
know
but-
either. I'm sorry.
Yeah. No, that's what I thought. I mean, so I think we sometimes have to be humble. I think that's what uncertainty factors are, humility in the face of uncertainty, of what we know and what we don't know. On the other hand, we also have these observations that in the real world, we are observing among human beings and children these outcomes. That is pretty clear. So.
Right. I just want to echo one panel member's previous comment is that the PD part right now is based on simple chemical reaction, PD, pharmacodynamic variability. But in the neurodevelopmental toxicity, the variability on the PD part, pharmacodynamic part, could be much bigger.
So let's go back to the model. Oops, I'm sorry.
Well, I just have a question. So you're saying that there are different mechanisms that could be responsible for those effects, and what are those mechanisms?
I'm not saying that we know the mechanism. I'm just saying that pharmacodynamic for RBC inhibition is relatively simple and straightforward. But the same cannot be said for developmental neurotoxicity.
But there is not even any speculation on what could be the mechanisms or any evidences?
As I said earlier, US EPA counted as many as 5 to 6 possible or plausible mode of actions.
And what are those?
They have really described in the TAC document, so I don't know if maybe there's uncertainty. I think DPL could probably answer that question with greater details than we can do.
Mm-hmm.
Coming back to the pharmacokinetic pharmacodynamic model and the point of departure, which has to do with 10% acetylcholinesterase inhibition, and that's what that whole model is constructed around, right? And you're saying
Correct.
that we're using cholinesterase as a surrogate for something else that we're not, cholinesterase inhibition as a surrogate for other effects, right?
Correct.
And you've come up with some uncertainty factors for they've come up with an uncertainty factor of 10 for neurotoxicity, and you don't disagree with that uncertainty factor. But isn't the net effect of that saying that your point of departure is 1% cholinesterase inhibition, essentially? Or am I being too primitive?
If there's a linear correlation.
Well, what does the model presume?
I think DPL probably can answer that question, what the 1% point departure would be
Well, okay.
from the model.
DPR who's the modeler? Yeah, can you chime in and say whether that model, which was developed by the EPA, I guess, and then refined by you, assumes a linear response for cholinesterase inhibition?
Okay, so let answer the question. I mean, back to the notion that, anything less than 10% cholinesterase inhibition is considered unreliable, meaning, technically we cannot achieve that. I mean, in terms of the model, you can actually go down to, well below 10%, whatever percent you want. But what would be the biological meaning attached to that, the model value of, 3% or whatever percent, below 10%?
Well, let me play it out for you. So you've got all these animal studies. Now you've got 7 or 8 or 6 or whatever it is, reliable animal studies with the endpoint of neurodevelopmental toxicity. And you have the dose that the animals got. And for some of those studies, you have multiple doses, right? And if the relationship between dose of chlorpyrifos and 10% inhibition is such, we heard many times 1 milligram per kilogram was the amount below which you didn't get 10% inhibition, right? But if it's a linear and a relationship, and if the animal models of neurodevelopmental effects show that your low L was 0.1 milligram per kg, and if you thought it was a linear response, then 0.1 milligram per kg would be a 1% acetylcholinesterase inhibition, and then you could adjust your pharmacokinetic, pharmacodynamic modeling to give you a point of departure of 1% cholinesterase inhibition, if it were linear, of course, which I don't know that it necessarily is. But have you thought at all in that direction? Because I'm trying to think about how, and I'd still say there's a lot of uncertainty, so I wouldn't do that and then do away with your uncertainty factor of 10, although you may say, I think it can be 5 now because we've taken part of it into account by doing this. But is that a route because otherwise I'm trying to figure out, even if you decide to use neurodevelopmental toxicity as your endpoint, how are you going to make that operational? And in terms of this is a side comment, but in terms of this back and forth about the epi studies and we can't get the dose response from them, in fact, if there were no epi studies, or if all the epi studies were negative, I would say there's enough animal data to have that be the endpoint. I think the epi studies are kind of the icing on the cake. I wouldn't try to use them for your dose response, but they just underscore the biological plausibility of all the animal studies. And add uncertainty because humans are likely to probably be could be more susceptible, and certainly what they're measuring in the rodents is a more blunt measure of neurodevelopmental toxicity because they're not looking at the SAT scores of rats,
If I may, and if it would please the chair, this was the opportunity for Oia to present their findings. DPR is more than happy to come back and spend the rest of the afternoon till midnight, if it pleases you, to talk about it. But I think it might muddy issues if Oia is asked to respond on behalf of DPR and vice versa. So if we could allow Oia perhaps to finish their presentation and then invite DPR back for questioning, we'd be happy to do that.
I think the idea that it's a great idea, and I think what we will do is we can wrap up this part of the presentation, and then have lunch. We'll take a 45-minute lunch recess, and then give DPR a chance to, because they've gone over this in detail and had given us their responses to these findings. I think,
And we have some good tables to show too.
and we have tables. All right. So let me ask you, are there any other questions that we need to address directly to OIHA on this? Jesus?
Well, my comments is not to OIHA in particular. It's just that I have the time limitations, and I will have to leave and just like around in around 20 minutes. So I will not be here for after you reconvene from lunch. So I don't know if maybe I should give my comments and to the DPR and now, and I will not be able to be here too for
Yes,
the discussion.
let's take advantage of the fact that you're here now. So go ahead.
Right. Will they be able to answer, or will you, or they just comment and when you come back from lunch and then you let me know? I just go. Okay. So
these are questions of a DPR or for OIHA?
So it's mostly for DPR.
Okay. So are there any questions for OIHA? Okay, thank you.
Okay, well, thank you very much. And Shelley, if you wouldn't mind. All right, go ahead.
Okay, so so this is a very, very extensive presentation based on large amount of evidence, and the majority of my comments will go or are focused on the pathogenesis and on the the toxicology. So you've done a pretty good presentation in the document about the what the compound is and the various characteristics and focus on the mechanisms, and is perhaps a little bit weaker, perhaps because there is not most of the work and has shown just the effects that that it has on the cholinesterase and activity. And there will be ample discussion, I'm sure, in the remainder of the afternoon. You will have already witnessed some on how to translate these and how to extrapolate from these and effects into the actual health effects that you may have. However, it strikes me that if there is a possibility that there are health effects and that can be induced even in the absence of demonstrable inhibition of the acetylcholinesterase, there got to be other mechanisms, and as those and 5 other mechanisms that were mentioned in the OIHA presentation. And so I've been doing some searches, and I've been finding some alternative mechanisms, none of which were mentioned in the document. So I could suggest that one of the things that could be done to improve on the comprehensiveness of the document is of including these other pieces of evidence. So the acetylcholinesterase inhibition, again, is very well documented and presented, but there was no mention about other effects, such as effects on oxidation, on oxidative stress, and effects on the peroxidase activity. There was a good presentation in between the relationship of the peroxidase 1 as an enzyme that catabolizes the compound. However, it goes in both directions. Not only the peroxidase and detoxifies and this compound, but also this compound can alter the activity of the peroxidase 1. And there is a paper and an abstract rate, so that in animals it's been described that there is a decrease in activity of the peroxidase, and after just one or low doses or low concentrations of it. that is rather important because the peroxidase 1 inhibition or the inhibition of its activity can result in a variety of different effects. For example, in knockout animals and for the peroxidase, so there's been descriptions of an increase in atherosclerosis and increased cardiovascular disease. So one of the things, and that has been well shown, it's been shown in a Nature paper many years ago. So I did some other sessions. So if that is the case, could it be that in addition to this neurodevelopmental inhalation and the dermal effects and that you mentioned in the document, could there be other effects that perhaps are not through the cholinesterase activity inhibition, that through the increased oxidation or through the inhibition of the peroxidase 1 activity? And it turns out that there are reports. So there are reports on cardiovascular effects, and there are epidemiological studies where it shows that there is an association of pesticide use with a myocardial infarction. And in one study that was published in 2010, and I can send you the references because those were not mentioned in this presentation. Although there was no overall association with the use of the 27 pesticides and that were included in that document. In that study. It's a study that included over 22,000 women farmers or spouses of the farmers. 6 pesticides were associated with myocardial infarction. Out of the 6 pesticides, this is one of those. The odds ratio was like a 2.1. So it was not insignificant. And they couldn't really conclude at the end because they said that they didn't really have the power to specifically determine like a risk and attributable to the specific pesticides, but that it was highly, highly suggestive. So that goes together with other papers where they have shown effects on lipids. So just one single dose of this compound can increase, and the lipids can increase apparently the cholesterol or triglycerides. I didn't have access to the paper, so I cannot really say what is specifically. There can be also increases in the glucose. And there are papers that to talk about an association with diabetes, either that it increases the risk of the diabetes or the diabetes could be increasing the propensity of developing effects. And there is a paper from Beata in our panel where it shows also associations with Parkinson's disease, and I think that she will be discussing that and with more details. So I guess that my recommendation is just to try to broaden, the consideration for all the different factors, to broaden the pathogenic mechanisms, to broaden also and include like the other potential and effects in addition to the neurodevelopmental. And I don't know if that will play a role or a factor in your considerations of the uncertainty factors, because if there are other effects and other mechanisms that we cannot really account for, that maybe the amount of the evidence is not enough to take them into consideration to regulate based on those, but it does add uncertainty. It does add uncertainty, and it adds, well, it could actually be that this is more toxic than what we think it is, because we're just not considering the full spectrum of the effects and that it could induce. So that will justify maybe keeping a large uncertainty factor. And the very last comment will be that it seems that there are some individuals that will be more susceptible than others, and you mentioned some in the document, and it has to do with individuals with different polymorphisms for different genes, and very specifically the peroxidase 1. And so individuals with the polymorphism or the genotype and variant that confers a lesser activity, so may have an increased susceptibility to develop various of these effects.
Thank you very much for those comments. And for the sake of brevity of a 278-page document, we did try to narrow the focus on the acetylcholinesterase. However, we have edited our documents, and in a previous version or previous draft, it was internal only, we did have a large discussion of an AOP. And if you have a few minutes before you leave, I would like to invite Dr. Marilyn Silver up here to talk a little bit about those pathways that she's been looking at, including endocannabinoid. Now, this was specific to developmental neurotoxicity. However, it gives a window into the complexity of this chemical and how the biological systems are interacting. You can just talk about the AOP. Oh, okay.
My name is Marilyn Silva, and I'm in the Human Health Assessment branch with Shelley Duteau. And so I think the most important, let's see, let me get this a little bigger here. Oh, okay, right. Okay, because I don't have much time and this is very complex, I would take a look at this box down here where you have your key events going on. And, and if we go through this really quickly, first you have, and most important, you have a disruption of the endocannabinoid system by chlorpyrifos. And then this leads to the various other key events, which ultimately ends with reduced connectivity and functionality of neural networks. And so there you're going to have your neurodevelopmental effects. We focus on the diagram on the left, and you'll see chlorpyrifos would act at the CB1 on the far left and ultimately inhibit the enzyme MAGL that breaks down the endocannabinoids. So the endocannabinoid buildup in the synaptic area will inhibit the release of neurotransmitters. And therefore, you have inhibited function. So we're talking about the GABA system, acetylcholinesterase, NMDA, the glutaminergic system, dopamine active transporter, and serotonin. So you're going to have an inhibition, I mean, in a nutshell, you're going to have inhibition of release of these neurotransmitters, which are going to have a long-term effect on downstream with your neuroprogenitor cells, your neural networks, and ultimately neurodevelopmental or neurological effects.
Thank you for that, Marilyn. And also, we have found just in discussions of other papers that they have mentioned potential metabolic mechanisms, including neuropathology target esterase, monoacylglycerol lipase, fatty acid amino hydrolase, and then other non-metabolic mechanisms, including, as you mentioned, oxidative stress, disruption of neurogenesis, cytotoxicity, disruption in cell signaling, altered nuclear transcription factors, altered neuronal cell interactions, and then you have the environmental factors, which we tend to not include, although weight of evidence would make us want to look at those, including whether there's combined chemical exposures, the health status of the mother or the individual, infectious disease, heavy metal exposures, and social determinants of health. So we're looking at an incredibly complex system, not only in neurosynaptic junctions and in the brain, but also in the environmental milieu that a person is in. Because we are a regulatory agency and we have to come up with a regulatory target number, we can only, for a risk assessment, look at a single chemical at a time. That's the system that we operate within. So unfortunately, we can't look at multiple OPs or the interaction of OPs and carbamates or any of those. So if we look specifically at chlorpyrifos, though, the world is just opening up in the potential pathways. And the AOP has definitely not been completely elucidated. This is from a draft publication that will be
has been
going.
accepted.
That's been
and-
accepted.
yeah, and is in revision.
But your points about multiple mechanisms and known unknowns and unknown unknowns is right on point. And did the selection of an endpoint that we did and the addition of the uncertainty factors to that, does that allow for enough protection for whatever might be happening with this compound at whatever levels as the target tissue dose?
The chlorpyrifos auxone inhibits esterases, and we know that esterases are about 11% of the total genome in humans. So, so with that, we would be expecting many pathways affected.
And in addition, the inhibition of the cholinesterase in the brain is going to lead to a buildup of acetylcholine, hyperstimulation of the entire cholinergic nervous system, and all of that, which then can lead to, tissue damage and a number of other things. So, I mean, there's a real link to, the inhibition of the cholinesterase, as well as all these other mechanisms. And I think it would be very helpful, just leading with where I would be going in our discussion, to improve the document by actually putting some of this stuff in the front end, so that there's a real rationale for why are we looking at, cholinesterase inhibition, and how does that link to the developmental nervous changes and nervous system changes. And I think that would help too. But anyway, I think this would be a good time for us to take
Does Jesus have anything else you wanted to say before you run off? You don't have to. I mean,
No. I guess that I understand the decisions that you made in terms of, okay, so you're choosing on the neurodevelopmental effects and you're choosing on the cholinesterase inhibition to make your decisions, right? And use the modeling and all the data derived from there. But that doesn't impede, at least, perhaps in the background or in the introduction, mention all the other potential. I'm not talking about interactions of the chlorpyrifos with many other compounds. I'm just talking about chlorpyrifos and the way how it acts. It induces all these effects and the variety of effects that it can induce. And if one of the things that we're talking is about uncertainty, well, how much uncertainty to say that we will be regulating based on a mechanism that we know that even if in non-existent, the chlorpyrifos could still induce some effects. That is a lot of uncertainty. So I think that and rather than just considering like a narrowing or decreasing the uncertainty, the question is that should we increasing the uncertainty factor even more? Because I think that we're not including other things that could be potentially as important or even more important. Maybe because they just haven't been studied enough, or maybe because they're not as important as we just don't know, right? So, but at this point, I already made it, and so I. If you decide to include it, so I can provide some of the references in some of these searches.
Yeah, we'd very much appreciate that. Thank you.
I just want to express gratitude for those comments, because I mean, this is stunning. This is really interesting, but this is still all that one pathway. And Jesus is also talking about totally different pathways, right? They're right, and so I don't want to lose that point of his. I mean, I think this is great that this points out all these other things that you didn't do, but right. Well, right, but it's all coming through. I understand that it's initiating all from the cholinesterase.
No, it's not.
No. Okay.
That's the whole point.
Okay, I'm sorry. Then I misunderstood that. I
No,
thought
no,
that was the
this
first
is-
step.
It's
Okay.
related to the clinical endpoint, if you will, is neurodevelopmental
Right.
toxicity. But this is underscoring that can be independent of the acetylcholinesterase
okay.
inhibition, and that's the biological plausibility of seeing effects at levels which we have reason to believe there is no acetylcholinesterase inhibition.
And so Jesus has given us other pathways as well.
Other endpoints.
And other endpoints, both, right.
I think oxidative stress is here.
Yes.
I missed it then. It was just, it was a little
Oxidative
hard
stress
to
is there.
take in.
But Dr. Hammond, your point is well made that if it's cardiotoxic or lipogenic, there are outside of these enzymatic pathways and signal transcriptase pathways that there could be other phenotypic expressions of toxicity that we can definitely look at. And as I mentioned earlier this morning, I did look briefly at spermatogenesis, which is not developmental, and it's repro, so it's different. And the effect was 35 milligrams per kilogram per day for spermatogenic effects, which is well above the effect for acetylcholinesterase. So looking at that one aside, but what are the sensitivities of these other endpoints? And I think it would behoove us to look at those.
Yeah, well, I wasn't impressed and in the document as well as in your response now, on the many different receptors that are can interact with the chlorpyrifos. So you talk about FXR, you talk about an LXR, you talk about a lot of nuclear receptors, and I'm saying, wow, all these receptors are very much involved into carbohydrate metabolism and lipid metabolism and a lot of systemic metabolism. Why we don't have much of many, more evidence of it? So, and that's why I started doing the searches, and I found some references with animal work and some references with human. But I have to say, yes, that there are few. There are few. The bulk of the evidence goes into the neurodevelopmental. Could it be just, and again, that they haven't looked at it? So, for example, you cannot see what you don't even look for it. You have to have some sort of like a educated mind, right, and to discover or
And if we had 5 more years to work on this document, it would be wonderful. We could always retire knowing that this was.
But you can certainly take into account at least what we know and what is being published. So even though the bulk of the evidence is on the neurodevelopmental effects and on the cholinesterase activity, the few pieces of evidence that are saying that there is a whole lot of other mechanisms and effects that haven't really been studied enough could be at least be mentioned and could be taken into consideration, perhaps at least to substantiate the large and uncertainty factors that we are considering.
All right. I think this is a good time to stop. We'll have a 45-minute recess and reconvene at 2:15. Good afternoon. I'd like to call this meeting back to order and continue with our discussions. So if everybody can get their seats. The next item on the agenda, we're going to have a response that's been prepared by DPR regarding the findings that we have presented earlier. And then we'll move on to discussing the charge questions. So, Dr. Depot. Sorry.
Well, I apologize. I didn't realize that we were expected to give a formal response to OEHA's findings. We have a document that everyone released, but we did that we released to the panel, but we didn't make a slide presentation particularly about our responses.
Well, what I think would be helpful is there have been a couple of issues raised,
sure.
And I think to give us just a feeling, nothing how you intend to deal with that, just as an overview. I know you've written up a very detailed discussion of it, but sort of
okay.
The high view.
Okay. Well, 2 points kind of bubbled up to the surface in the conversation at the end of the morning. One of which was on the differences on breaking down the uncertainty factors between OEHA and DPR. So I think if we could start there, that would be a really good place to start. And then we can also clarify where that extra 10 comes in as a toxic air contaminant. So let's start there, and then we can move on from that. So I'll turn it over to Svetlana Koshlokova. So the table we're showing on this slide right here is, Svetlana, if you can do slideshow, and that gets rid of all the stuff around. It's on the next to 71, exactly. This was actually a table 4 that we pulled from DPR's response to OEHA comments, which was released in August 2017, just ahead of our PREC meeting. So we don't want to speak for OEHA, of course. We pulled the explanation of their parts of their uncertainty factors from their comments to us, but we certainly don't want to speak to that. What we can do, if Lori or David are willing to, is to come up and they can speak on their numbers. We can speak on our numbers. But if we start, I'll have Dr. Koshlokova start on speaking to our numbers.
So just show the...
So I think that's an idea to have OEA talk to, and what I would prefer, or go back to your slide, because your slide and OEA's slide are the same in terms of the numbers. Maybe to go through these one at a time and hear from you and hear from them about each one, and then have some discussion. Because I do think that this is like this is probably the biggest single issue in the report, is what these numbers ought to be. So, and the other thing, by the way, I will use my prerogative as the longest serving member of the committee who has been on it forever. This process of discussion, rather than having to worry about a lot of formal documents back and forth, is the way we've done these in the past. And then you get a transcript and get to try to figure out what everybody said after the fact. But no, I think this is like one of the 2 or 3 key issues. Okay.
And I guess let me ask Lori and David, before you talk to your numbers, and maybe we can start with interspecies and then interspecies. Would you like to have Randy come up and explain the 10X for the TAC? He's willing to do that if that's okay? Okay, good. I'm going to turn the floor over to Randy Sagawa then.
From the PREC presentation, would you like that one?
No, it's all right. Okay.
I didn't mean to cut Svetlana off, so.
Oh, that's fine.
Okay.
Yeah, so part of this discussion this morning was this extra tenfold, quote, uncertainty factor for toxic air contaminants, which adjusted OEHA's recommended total uncertainty factors from 1,000 to 10,000. That extra, quote, uncertainty factor is really not an uncertainty factor. It's not based on science at all. It's the legal criteria that DPR has for listing as a toxic air contaminant. So whatever total uncertainty factor you end up deciding, whether it's 100 or 1,000 or something else, then under the federal, state law, for listing as TAC, we add in an extra tenfold factor. And so if the margins of exposures exceed that extra tenfold, then we list it as a TAC. Again, it's entirely based on legal requirements. There's no science behind it. And so it really shouldn't be viewed as an uncertainty factor. Okay.
To finish clarifying that, but and also from the chairs, but what we should come up with as our recommendation to you is we think we should kind of forget about that legal thing, and we should just tell you what we think the number ought to be, and then you can go fight with the lawyers.
That's exactly correct.
Okay, great.
So this document that you see, so the document that you see on the screen is our response to OEHA's comments that Shelley mentioned. It was published on the DPR website on August the 15th. And so these are our responses to their comments to the 2015 RCD. This was before the bulk of the 2017 papers were published on developmental neurotoxicity. So if you go to page 8, we have this table that we composed comparing the uncertainty factors used by DPR and OEHA and recommended by OEHA. These uncertainty factors are for colonization inhibition as an endpoint, and based on point of departures estimated with the Pbpkpd model. Our reasoning is to the left in the blue highlighted section. For the interspecies, we are using an uncertainty factor of 1 based on the fact that those are human equivalent doses derived from a Pbpkpd model. This is a human model, and the outputs, the PODs, are for humans. For the interspecies factors, this is the variation in sensitivity between humans. We retain the default of 10. At that time, the model that US EPA used, it was developed by Dow, established a so-called data-derived uncertainty factors of 4 for chlorpyrifos and 5 for oxon, which would indicate that people can vary the sensitivity to cholinesterase inhibition in humans is 4-fold for chlorpyrifos and 5-fold for chlorpyrifos oxon. However, we had concerns about these DDEFs, the data-derived extrapolation factors, mostly because we did not, EPA at that time, we adopted their endpoints, did not utilize the pregnancy compartment of the model, so that life stage was excluded. And at that time, we had some concerns regarding the metabolic parameters and their variability. And the final uncertainty factor is a 10 for neurodevelopmental toxicity. This is to account for the fact that we're using an endpoint for cholinesterase inhibition, and we are aware of potential neurodevelopmental effects. As such, we evoke the uncertainty factor of 10 for this potential, neurodevelopmental toxicity that may occur at doses lower than cholinesterase inhibition. We did not have a lot of evidence where the neurodevelopmental effects may occur at what doses. At that time, we had one evidence from zebrafish studies. This was not a ToxCast or high throughput toxicity study. It was a normal lab test where they measured cholinesterase in zebrafish and also behavioral effects and malformations, and they found that those neurodevelopmental and neurobehavioral effects occur at about 10-fold lower concentrations than cholinesterase inhibition. So that was the reasoning for our uncertainty factors. Okay, so then since then we
In your tables of in the document, in the parent document where you had tables about neurodevelopmental toxicity, the tables at least seem to be limited to rodent studies. So the zebrafish data aren't included there. Are they included somewhere else that's just hard to see where they are?
They are in the RCD. Yes, they are included in the RCD. It's in the ToxCast section.
Okay, I understand. So that's probably why I missed it. But when one goes through the document, you see this section on neurodevelopmental toxicity. All the, rodents, and there's nothing about the zebrafish there at all. You may want to give a shout out to there's also zebrafish data which is covered in a different section or something.
We will ask Madeline to present a little later. We have prepared a table where we compare the developmental neurotoxicity studies, the newly published ones, as well as zebrafish on the next slide.
So let me just go back to the topic of the endpoint. Maybe we're too far down the road. Maybe we need to establish something else a bit more basic first. What do you propose as the endpoint for toxicity in your standard setting?
Specific to chlorpyrifos?
Yes.
So our assessment to date shows that the database for acetylcholinesterase inhibition...
I didn't ask what the database was better for. What do you think is the key toxic endpoint for chlorpyrifos? Not what can we model with acetylcholinesterase. What do you think is the target organ and target effect that is the public health concern here, the greatest public health concern? That's, I think, what we need to decide first, and then everything else is just tactical. Strategically, what?
So you're asking a question of a regulatory agency that is difficult to answer, because if we're protecting public health as we are, we have to refer our entire mission. And so what we have to do with establishing a regulatory target is have something that's scientifically justifiable and also defensible in court and defensible with the legislature. So we're not simply coming to this body to come up with a guidance level. We're actually going to be making regulation following this. And the weight of evidence that goes for that may have a different factor that we have to consider. So I cannot answer that question without talking about database certainties and database, just the volume and certainty of a database.
Yeah, I really think it's a little bit of an unfair question to ask them. I mean, they've come forward with their report, and I think it's our job to say what we think they should do. I mean, if you read it, I mean, as I recall, the very last thing is we're using this red blood cell acetylcholinesterase, but we recognize that the neurodevelopmental toxicity is important and thinking about it. And I think the question, the thing, and I agree with you, this is the fundamental question. And I mean, just to speak for myself, having, read all of this, talked to them a week or so ago, read the public comments, listened to the discussion this morning, I mean, I think that developmental and neurotoxicity is the more appropriate endpoint, and that you could develop a defensible document and a defensible position with. I think that you have, I mean, I think the document is actually well put together. I could follow it, which is like amazing, because a lot of this is not what I do all the time. And I think it's really a matter of judgment and emphasis. And I think that you have in the document, the information that you pretty much need to make case for developmental neurotoxicity being the appropriate endpoint from a regulatory point of view. I mean, I'd be interested in hearing what other people say. Now, just one more second. The next question after that is then how do you then convert that to a number that you can use for regulation, and that brings us back to this.
Thanks for restating it, because that's really what I was trying to add.
No, but the thing that was different, I think, just, because I was quiet this morning. My head almost exploded. I know. But everybody else was asking all my questions. But I don't think it's fair for us to ask them at this point what they think should be done. I think it is now our responsibility to say to them what we think they should do. Do you see the difference? I mean, I think they put forward what they think in this report. I think it's a well-written document, and I think it's been very fair in presenting the data on all of these issues. I think they were very responsive to the public comments, within the point of view that they ended up with. And I think the question we have to ask is, do we agree with the primary conclusion they drew, or do we want to recommend that the alternative pathway, which is outlined in the report pretty clearly, I think. So I'll be quiet now for 5 seconds.
I'll try to make this quick. To answer that question, and I agree. I think that this is these are the two primary questions that we've been talking about. I understand what you're saying about needing the strong database. I think you have shown using an outcome that you don't think is the prime outcome if you really had well, at least this, let me say, I think, all right, okay, so for myself, the red blood cell acetylcholinesterase inhibitor, I won't get that all word, cholinesterase inhibitor is, as an outcome, is less important, I think, than the neurotox developmental outcome. But there's a stronger database. But what I see is you can take the first outcome and you can do all your uncertainty factors and your numbers and work that out. And we have enough data to say that we're seeing these neurodevelopmental effects happening at lower concentrations. And so I think you make your strong case for outcome one, and in what the level would be to that, and then for sure that outcome two there's an uncertainty factor. We can talk about what that should be, but you've got an uncertainty factor, and you could start saying where can you work from there, but it's sure as heck going to have to be something way below the first one, because you already see that effect. So I think there's enough data, and that's how you would support it. Is you prove A really well, and then you say B is less, and then you work on how much less.
And again, because I was so quiet this morning, I don't think this is going to require a huge amount of rewriting of the report. Because I think you have a huge amount of information on just this point there, and it's very well presented. I think the stuff that you presented right before lunch, without getting into a whole, writing another 100 pages, I do think that strengthens the case quite a lot because it points to the biological pathways that kind of connect all these things. So I definitely think that's, and I thought you presented it pretty clearly too. I think that's worth adding, but I think the way that Kathy framed it is, I think, what the path forward is. If the committee ends up agreeing that neurotoxicity should be so.
So I'm actually really stunned to think that what I heard, and I might be completely wrong, is that you're basing all this on the acetylcholine esterase inhibition, more or less, even though you're presenting all this data, the ToxCast, the assays. And all of these assays are pointing all in the same direction, right? Receptors, pathways. And this is not that modern anymore. I mean, this has now been accumulating over a decade. That's probably why you were able to put this in here. It seems like you're ignoring the huge database that was created with these assays that all tell you something or parts of that big story that probably leads to neurodevelopment, neurodegeneration, something brain related. So I didn't really see how that was brought your overall ruling. You described it beautifully, and that's nice, and thank you. But how was that brought into the decision-making, the rule-making, or was that all knowledge you put in after you already had decided on these factors? It's not clear.
Okay, so just to clarify, I think, we chose an endpoint, which is acetylcholinesterase inhibition for this document. And then we justified what the uncertainty factors were to get to our reference concentration for an inhalation toxic air contaminant. And I believe if it wasn't clear in our risk appraisal section, we can certainly go back and make it more clear that to underpin a 10-fold uncertainty factor for neurodevelopment, that is underpinned by the AOP that is still at this point, also by the zebrafish data, and also for the epidemiology data. All of that goes into basing a 10x neurodevelopmental uncertainty factor where we are right now. The additional animal data and any additional epidemiology data that comes in could certainly strengthen that number or change that number. So hopefully we didn't discount it. Hopefully it was just a matter of not being as explicit as we should have been in the explanation.
So that sounds to me like you have 3 lines of evidence: epidemiology, the animal at very low doses, and the ToxCast assays, and all 3 of them give you a factor of 10. I'm wondering how that number is justified.
So our first risk assessment came 2015, so you understand this was years ago. So this table that you see is responding to comments to our 2015 risk assessment. Since then, we have made 2 more drafts and new information came. So in this particular case, at that time, the only information that we had regarding to how much lower neurodevelopmental effects might occur or from the zebrafish studies. We have experts in ToxCast. We have a winning group here, WIHA and DPR. And so we did explore ToxCast. We have a lot of pathways, but at that point, we're unable to quantify how these pathways, in vitro pathways, would produce in vivo human doses. That was at that time. Now, recently this year, Since 2015, there were 2 poster presentations from Dr. Carr's lab, and then several papers were published, most of them this year. All of the effects you see on neurodevelopmental toxicity, they're in 3 domains: cognitive effects, behavior, and motor activity. All of them occur in the 4 papers occur at 0.1 milligram per kilogram per day. Remember, the threshold for cholinesterase inhibition is 1 milligram per kilogram per day. So we are about at 10 in animal studies.
Sorry. So to come back to Kathy Hammond's direction of questioning, and to combine that with Beat's questions, seems like there are only 2 logical ways to go. One way to go is you say acetylcholinesterase is our surrogate endpoint, even though we do not believe that that is the mechanism of toxicity. But we're still going to use it because we believe our data is more robust. And we believe that at least that a dose of 1 is the lowest effect level. It's not a NOLA, it's lowest effect level, right?
There is one.
But we're still going, but if I understand it correctly, despite all of these correction factors, you're still using the point of departure as 10%. And then after the fact, you're doing a series of adjustments for uncertainties. But usually when we're talking about uncertainty factors, it's not like, actually, we're pretty certain this isn't the mechanism of action, but we're going to use this anyway. So that's what's not so attractive about using acetylcholinesterase inhibition as a surrogate measure. But if you are going to use it as a surrogate, you're going to have to assume that the surrogacy occurs at 1% inhibition or 0.5% inhibition of acetylcholinesterase. That's even as it's a linear effect because you're seeing something instead of at 1 milligram, you're seeing at 0.1 milligram. So I don't think you get there just by throwing in an uncertainty factor of 10. The other way you could do it, and I'm not so sure I understand why you can't do it this way, is forget about the acetylcholinesterase inhibition, forget about the Pbpkpd modeling, take the animal data that you have for neurotoxicity and either try to estimate a benchmark effect or a low L and divide it by 10 and use that and do all your calculations with that value, or at least compare them side by side and see what they look like.
Under that paradigm, you needed 10 for interspecies extrapolation.
Yes, you need a 10 for interspecies because you'd be using animal data. You'd have to...
Right, and I think that the 10 for developmental neurotox would then to 1, so it'd be the same number, same uncertainty factor.
I understand that, but
the same total uncertainty factor.
But since you'd be doing a low L, I guess, at 0.1, it's actually going to be 0.01, right? So you're going to get 10 more that way. So my guess is...
No, low.
Yeah, so my guess is come out, 60-fold lower or something, I don't know, than where you are now, something like that. But at least it'll be a bit more logical than saying, we know that this isn't the mechanism we care about, but this is good data for, doing our calculations. But that's kind of putting the cart before the horse a little bit, I think. It would be better to do the cruder, no L way. But you might even be able to have a model for what I want, where you do the doses and a benchmark thing that will work. I don't know if the data are robust enough, but I would at least try to go down that path, and then you're not forced to put yourself in this double bind that you're otherwise in, it seems to me.
But I think it's important to keep in mind that the inhibition of the enzyme is part of a mechanism and could be a toxic outcome. It's just the fact that other things are happening even at lower levels.
Yeah, the
So
target organ of
think
toxicity
I would
or
say,
the target health effect.
but to amend what you were just saying, I would not say that it's not the not a mechanism. I would say it's not the only.
Well, it doesn't appear to be the mechanism for neurodevelopmental toxicity since it's occurring at levels below which we would have anticipated there be any, and in which there doesn't appear to be any acetylcholinesterase in it. And I thought your figure, which tended to indicate that if you had to, someone put a gun to your head and choose the mechanism, you would say it was the cannabinoid mechanism. That was kind of what I took away from that figure, even though that there's some other possible explanations.
Well,
but anyway, we've
necessarily, this is the one that we have more information how chlorpyrifos works. It just the data is there. There is quite a bit of information on serotonin, hitting the serotonin system, oxidative stress, but this one is the one that we can make the adverse outcome pathway as a first try.
Yeah. Anyway, that's. Wouldn't that make your life easier, not harder?
It still would require an extensive rewrite of the document. That aside, we would be interested in seeing what this panel's recommendation and findings are to us.
Well, that would be mine. And why don't you speak next?
Well, I'm an epidemiologist. I just want to see all the human data acknowledged. And I think we already heard from my colleague that there might be other data out there, which because, we live longer, we have a neural network that is slightly different from all these animals. And there might be more chronic effects than we really can imagine. So we should at least point to them, even so we can't use them for regulations yet. But they should be acknowledged and taken into account, especially if then tox comes more and more of these pathways coming up.
It all comes to the uncertainty. Which endpoint selection would lower the uncertainty? Is it the well-docked cholinesterase inhibition that we know and then just add additional uncertainty factor to account for neurodevelopmental effect, or use neurodevelopmental endpoint, which comes with uncertainty because the database is not rich and they're not solid endpoints at this time? So we prepared this table per Dr. Glant's request. So I can walk you through. It's a little bit complicated, and I'll try to narrow down to the topic of the conversation right now, and then I'll get some help. So basically, it has 4 panels. The first one is the uncertainty factors, the route and duration, and then various exposure scenarios, as well as the sensitive population that we evaluated. So the second panel is in pinkish, and that's our current document, 2017, December. It shows, the first column shows Pbpkpd derived point of departures. Those are based on % acetylcholinesterase inhibition using the model. And we expressed everything as microgram per kilogram per day, so it's a little confusion about the unit. So remember, it's microgram, not milligram. So the second part is the reference dose or reference concentration, which we calculated as the critical endpoint divided by the uncertainty factor. In this case, for the model that we use for the endpoint, our total uncertainty factor is 10, based on 1 interspecies sensitivity, 10 for intra, and 10 for developmental neurotoxicity. So if you look at children only for acute oral, the reference dose would be 5.81 microgram per kilogram per day. We go down for acute, then we go down the road. And for children, the steady state, this is short-term subchronic. This is a steady state or equivalent to subchronic. Repeated dose treatment would produce a dose of 0.99 for 10% cholinesterase inhibition. This would be the dose. Nothing higher than this would be acceptable. And then we have the same for dermal. And then the same for inhalation. The one that's important is 28.5. That's the reference concentration, which for now is used as a target concentration. The panel in green is the published developmental neurotoxicity studies in animals. The point of departure, there are 4 or 5 studies. They all come with a NOEL of 0.01 milligram per kilogram per day, which would be 10 microgram per day. If you compare, now, those studies are done through different exposure regimen. So it's one of the studies is a single dose, acute, and then the rest are repeated treatment during gestation, during postnatal period, or combination gestational postnatal. So and it's all oral. That's a very important point. We only have oral data, oral studies. So again, neurodevelopmental point of departure will be applicable for both acute and short-term duration. And perhaps even longer, because at least in one of the studies, treatment is one during the postnatal period and then assessed when they become adults. So then we compare, if you look at the steady state, cholinesterase inhibition-based reference dose compared to the one that would be coming from developmental neurotoxicity studies, the difference is tenfold. That's where we come here. If we compare this reference dose to the acute number for cholinesterase inhibition, the difference is 58-fold. And then we go on to dermal and inhalation. We do not have route-specific data for neurodevelopmental effects. Therefore, we have to do some conversions using default assumptions of what the breathing rate is, and body weight for children. And then in the case of the dermal, we would need to have some idea of the dermal absorption in animals. So using 3% dermal absorption in rat, which is a published value in one of the US EPA's documents, 2011, the last document that used animal data for point of departures, we calculated a dermal POD based on the developmental neurotoxicity study as 3.3 microgram per kilogram per day, and this is going to be 403-fold lower than the corresponding one from cholinesterase inhibition. But again, this is based on route-to-route extrapolation. And for inhalation, the important number that we have, this is the current one based on cholinesterase inhibition. And if we use default rate of 0.33 cubic meter per hour and body weight of children 11, then we come up with this number, and it's going to be 1,000-fold lower. And then the third column is that Dr. Glant's request to also include the USCPA assessments using epidemiology study. So we selected the November one because the previous one in April did not really establish PODs. It was based on internal blood concentration, the concentration of chlorpyrifos in the cord blood. And so this one had the reverse dosimetry established reference point of departure. So USCPA's risk assessment did not deal with acute toxicity, so everything was done on a steady state base. These are the numbers, and this is how the difference would be compared to our current document.
Very helpful. It's also quite reassuring, actually, to see that the pathway gets you a bit closer to the EPA estimates, but without having to jump through all the hoops of their complicated model that was hard to decipher based on what you've said.
And it hasn't been vetted, hasn't gone out to scientific review as yet.
Their model.
The question for clarification, so all of these models steady state assume you're only exposed through that one route, right?
Yeah, that's correct. Because that's how we model it based on, it's a route-specific POD.
And for being a TAC, although I know in the past when pesticides have come in front of the SRP, the panel has considered either aggregate exposures or different routes of exposure other than inhalation. We're looking at toxic air contaminant, the route of exposure that we should be concerned about for this particular case should be inhalation.
Just to that point, I think, but we're also interested, reason the others are important is if the inhalation adds to and takes it over the magic line.
Absolutely.
So
and
that's why you have to look at all
absolutely.
those. And you did.
And I think, and maybe Eric or Svetlana can add to this, but the reason why a steady state exposure that we've modeled in our risk assessment is so important is twofold. One is that it's a departure from what DPR has done in previous risk assessments in that we are setting up a population that's already at risk. We're looking at a population that has been exposed according to the EPA model in 2014 for 1 hour a day for 21 days. And as that decreases the amount of cholinesterase available, the baseline inhibition kind of bottoms out or reaches that 10% steady state. And then we look at what happens to that population with that DEC when they get another hit. So we're not looking at a healthy population, we're looking at a population that's already affected.
So that, to me, that's a new point that I didn't realize, that effect is long-lasting. I didn't know how long the inhibition lasted, so that is a subchronic effect. Okay, and I was thinking it was like a day or two.
Subchronic,
no.
right. And I know that you've seen many charts in your lifetime about occupational exposure, and that's why, we talk about people having headaches over the weekend, and then they come back to work on Monday and they feel great. With carbamates, that's definitely the case with cholinesterase inhibition, where they get a hit and then they rebound. But this is a successive decrease in the ability of the body to handle the insult, and it goes and goes and goes and goes until it bottoms out.
Gotcha. So then, but I started out with another question, and that was in the green set of columns. For the female, is that not applicable? I'm not sure, NA or not available, but I thought, this is the animal studies, and I thought the animal studies included exposing the mother when she was pregnant and the gestational exposure. So I think the exposures we worry about. So this is a wonderful table. This is great. So let me be really clear. So I'm going to pick on the little thing.
I want to make 2 points. The animal studies are few of them, and usually, so those are published studies. Those are not registered and submitted studies. So for under FIFRA requirements, there are all kinds of examinations that are done on the moms. Published studies do not follow FIFRA guidelines. Therefore, at least the one that I'm familiar with the most, the 2017 study by Silva, mothers were treated late gestation, and then all of the evaluations are done on the pups.
I understand the evaluations being done on the pups, but I was thinking that the exposure would be in utero.
Yes.
And I think that that's one of the exposures that we're concerned about as a society.
But the effect is behavior of the pup. So yes. that's the.
Oh, so you're talking about where the effect was evaluated, not when the exposure occurred.
Right. They didn't do gross morphology or anything in the females. Although there was the one study, who did the adults with the sub-Q? Mueller did a study on adults, male rats, sub-Q, and there were effects in that species. But it's really hard to take a sub-Q, dose, subcutaneous, and inject the skin and make it relevant to a human exposure out in the real world.
Yeah, no, that's fine, no.
And
I just don't know
remember-
the little short words.
But there's a point that in Kathy's question, which is this: the fetus is exposed from the air going through the mother, right? Right, for the respiratory.
Mom is gavaged or treated.
No, but assuming that mother is not gavaged, but mother is outside next to the golf course and is inhaling in her last trimester, right? And in your adjustments, of course, you use the breathing rates of 1 or 2-year-olds in this class, right? But I don't know the answer to this question, but how are you going to take into account that what really matters is the breathing dynamics of a woman in her last trimester, when this in utero exposure occurs, that may be the most important exposure in terms of safety?
I mean, I guess the thing is that the table, as I said, I was thinking of it as at the exposure level. You could make this table, though, where you say which was exposed, the mother, the female who's 13 to 49.
We can pull
but the outcome would be at the top, that it's the DNT outcome.
Right.
But which would be on the children.
Right. So I think it might mean that you should consider I know you've only considered here for this quick thing, children, but you might actually, for the neurodevelopmental endpoint, need to consider the women of childbearing age as well, because even though the end point of effect wouldn't be them, it would be their fetus.
So are you saying that if there is a alteration in behavior, for example, anxieties that is increased in this study, would we consider that level that causes anxiety as a effect
No,
on the mother?
I think
let me
there's
try
a
to say
misunderstanding.
this, okay? Because I'm sorry, Joe. I'll do this quickly then. What I'm trying to say is the outcome that we're interested in is just some neurobehavioral effect at age 1 or 2, right, for the children. But we would actually look at 2 levels of exposure. The animals, you said earlier, the studies had this. Some animals were exposed only while the mother was pregnant. I mean, this is the animals. I don't have the right words. The dam, I guess. And is exposed while she's pregnant, but that's it. And then the offspring are not exposed. It's just an in utero exposure and no life. Others were exposed only after they were born. And then a third group were exposed for both. And I think that's what I'm interested, the outcome that you're going to look at would be, I'm just making this up, like the 2-year-old neurobehavioral outcome. That's the one outcome. But those 3 different exposure scenarios are really important and have different implications. As Paul says, the inhalation rate is going to be very, if you're talking about an in utero exposure, it's the dam or the mother's inhalation. So that, I think, is the way one would want to look at it.
Can I make a comment? Yeah, I totally agree with that, because fundamentally it's the dam that is exposed to the Chlorpyrifos in the air. So technically, when you take that point departure and convert it, it should apply to the female 13 to 14-year-old, which is the women of childbearing age, not applying it for the children, because the idea is that the reference dose would protect the pregnant women or protects the fetus.
And I also wish that we had inhalation exposure data in animals. It's very hard to expose pregnant dams. So the numbers that you see, the inhalation number that you saw on the table, is a route-to-route extrapolation. All were either exposed by gavage, so the tube down their throat, so an oral exposure, or subcutaneous injection. Inhalation is a very rare thing for us to find. In fact, we're working with one of Dr. Buckpitt's colleagues at UC Davis on doing one for Chlorpyrifos, and the amount of work that goes into creating an inhalation chamber is intense. So I wish we had more, but so as Dr. Kolshlokova was mentioning, there's a lot of assumptions underlying around extrapolation from going, okay, if you're giving a bolus amount down a tube into someone's stomach or this rat's stomach, what does that translate into airborne exposure? I mean, if we added the inhalation rates, which do change during pregnancy, you know, we could modify that somewhat by what our assumption is for inhalation. Instead of 3 liters per minute, maybe it's 3.5 liters per minute or whatever it is.
Yeah, I really enjoyed reading the document. It took me a while to get through it. It's a very long document, but it's a nice document. I would second what was said earlier, or third it maybe. I would just write in your document, there's 2 for neural damage, and, you know, at least 2. And one is the classical nerve gas inhibition of acetylcholinesterase one, and that one you've got covered very well. It's very defensible scientifically and in court. You won't have any problem with that. And then just write number 2. And number 2, you know, the neurotoxicities, something that probably began to be recognized 7 years ago when I was on the science advisory board of EPA. We put a lot of pressure on them to write a review article on that. And then it began to show something from a review article Bob Sanawani wrote. And now, you know, it's beginning to become more clear, but it's still not very, very clear. And if you leave that in there, then you can, that you're trying to protect the public health, that's defensible. And you're pretty sure that there is a mechanism that's just not mature enough scientifically that we know what that mechanism or mechanisms are. And then you take your stab, as was suggested earlier, at what kind of calculations that gives you. And when I first looked at this and the response Dow was giving you and stuff, I thought, yeah, it's going to be a little shaky to defend this. You'll have some difficulty, but you say, this is what it is, this is what we know, this is as far as we can go, and that's it. But we've tried to be health protective. We're pretty sure there are things like Parkinson's and other neurologic damage, the tip of the iceberg here with this pesticide, and probably with many more that work through this type of mechanism. And I think you're obligated to put that mechanism, you know, whatever you know about it in there, and do exactly what was said. Make your calculation, your second calculation based on that. And that way, if that gets knocked down or something like that, then you still got the other one to fall back on, but you've done your best to substantiate this other one, the newest one, because it is more health protective, and that's an honorable thing to do too. So I think I would put them both in and substantiate them as best as you can, and draw them, give them names, you know, that they have different mechanisms.
I mean, I agree with that too, but I just want to come back. I don't think this will take totally rewriting this document. I mean, I think you pretty much have everything you need. I mean, I didn't know a damn thing about this till I started reading it, and my wife every night, what do you do? I'm reading about this chemical where I can't pronounce the name. And so I really think these are matters of sort of framing and emphasis. I like the way Joe's suggesting it, and I mean, you've done the calculations in a week, so that's good. I think this is much easier to follow, you know, the green column than all those gyrations the EPA went through to kind of back calculate things and do handsprings. So I mean, again, having been on this committee forever, I mean, I think, you know, seeing things that we've dealt with and through, and where people, you know, ARB got sued over them, they won all the lawsuits. So I think that, you know, our job is to help give you that guidance, and I think you're getting a pretty strong consensus. And I think you've got it pretty much here. And I don't hear, the last thing is, I think, at least in listening to people talk about this, I think the approach that you took, and by the way, one thing, if it isn't obvious to anybody, which the DPR told me on the phone, is that these animal studies in the green weren't available when the original document was written. This was information that came available as the document was evolving, and so it's just a matter of now that it's there, making the full use of it. And the other thing that I found interesting in listening to this is, like, you have your uncertainty factors up there, and nobody's complaining about them. I mean, maybe I'm missing something, but, You know, I think by using the more direct endpoint, that makes the uncertainty factors less controversial, unless I'm missing something. Kind of the way you ought to go with this.
Well, this is different than what I learned in class from Dr. Hammond, and that is if you have human data, use it. So now we were using a human-based Pbpkpd model, and now we're saying, it's not so great. We're going to go back and use animal data.
I actually thought
I needed to give her a shout out about this.
Well, but the thing is, though, I mean, and it is a very nice model, and I thought the sensitivity analysis you did in it and all of that was actually quite impressive. But it's like the facts kind of overcame the model, and now I think the big thing that I see the epidemiology into this is that it shows that these neurodevelopmental effects are very important and occur at lower levels than the red blood cell effects. And you saw that in the zebrafish, and you have the animal studies. And so I think that, I mean, the way I read this is that the epidemiology justifies the change in the endpoint, even though the animal studies that you have aren't as extensive as what you have on the red blood cell outcome. They do give you enough to estimate a dose response. And you're selecting them because of the human epi. That's how I read this.
And I teach occupational epi and environmental epi, and I know where you're coming from. We are all coming from worker cohorts where there's high dose exposure in humans that we can characterize pretty well. To characterize pesticide exposure lifelong, even in farmers, is extremely hard. And to characterize anything in children is also very hard. And here we had an opportunity to take blood samples at one point in time and urine at one point in time, but then extrapolate out to neurodevelopment. That is not the greatest data, but even so, it's really not the greatest data, but it may not be good enough to justify everything that you want for regulation, but it points you in the right direction, right? And of course, we need more data from the humans, but then the animal study comes back in and says, hey, we actually can do this now. And that's the beauty of the animal studies, going back to them with the dosing.
And I think that even though there's a paucity of animal data right now, there are some FIFRA studies that we could maybe go back to, and I think maybe either Svetlana or Marilyn can speak to briefly. But there was a registrant submitted study that was a developmental neurotoxicity study, right? I know that they did not measure Chlorpyrifos concurrently with the DNT effects, but Hoberman?
Yes, it was
Hoberman and those are the kind of studies that we mentioned at the December meeting, where we have a volume of data. There's a 100-page summary report and then 1,000 pages of individual animal data. Those are the kind of data that we would really like to be able to get our hands on and do some individual analyses. Like Blanc was suggesting that we could do our own 95% confidence intervals or covariance or whatever we want to look at, but on a published study where we don't have individual data and they're only publishing summary data without supplemental information, it's kind of hard to get to the detail normally used as a standard for our level of data precision and accuracy for our risk assessment. But I'll let Dr. Silva talk to this one registrant submitted study.
Oh, well, this
The interesting thing about this, the interesting thing about it's the first line. Now all these effects I've described here are occurring at the low L, and the low L for all these is low L for behavior. The Carr and Mohammed studies were all done with 0.5 was the lowest dose tested. And like Dr. Glantz was saying, it's so new that I was presenting a poster at the Teratology Society. Happened
to run into this poster about chlorpyrifos that was Dr. Carr's. And that's kind of what, you know, sort of got the idea started. But so 2015A is a poster, and Mohammed et al. Is a poster, but they're the same laboratory. And he used postnatal day 11 to 16 as his time of dosing, because he said that was corresponded to the human postnatal period of brain development. But as you can see by lines, there's different strains of rat, mouse, all different treatment periods. There's the DNT, the standard GD through GD6 through lactation day 11, and Ragdolly rats, the pups treated postnatal day 11 to 16. Wistar rats treated during gestation, and male pups were tested postnatally. Wistar rats. So there's a whole gamut of developmental periods covered. It's not just in utero treatment that's important, because you're getting a lot of things happening with postnatal exposure as well. And then the bottom line is treatment in adults, and you're still getting the same low L. Yes, Hoverman is the DNT study.
Yeah, I don't think the point about gestational exposure was that you shouldn't develop a value for 1 to 2 year olds. It's just that you might need to develop a value for the childbearing age women as well. Two separate estimates, because I think that was really more the point of that. And I think that as you write your narrative to justify the tenfold safety that is interspecies, I think you need to emphasize that the things that you can look at in a rat are not as subtle as what you could look at in humans. Right, I mean, that's the main thing. It's just in terms of neurobehavioral outcomes, it's not nearly as subtle. And then I think for the intra-species tenfold factor, there are a variety of things that would make you, consider vulnerable positions that would justify that without much trouble given the endpoint.
Well, I think what's interesting about this too is that so many different vehicles were used, and different laboratories and different treatment periods, but you're still coming up with this number, which is
Actually, I'd be a little careful at you're right.
No,
but the reason the low L's coming into the same number is that that's the lowest level that everybody tested.
That's right. But
it's very consistent. If you full data set that we could conceive of, it might well be they'd have different numbers where they happen.
Yeah, and I think Carr's lab had 0.5, but who knows what it could have been. And there was one study, I think it was Silva, that actually had a NOEL 0.1, and so some of these studies did not have a dose response for those effects, and that's where I think the effects may be occurring at the level of the endocannabinoid before the acetylcholinesterase inhibition occurs, and so you could be having low things neurodevelopmentally happening before the
another argument, by the way, in terms of the interspecies safety factor is that actually remarkably don't have any kind of non-human primate experimental data. You might want to allude to that one Dow study of rhesus monkeys,
rhesus monkey.
For 6 months. That's the only primate study that that I saw anybody ever refer to.
We're trying to get that. It's actually a medical college, Albany Medical College report, which was never published.
Yeah.
But we're trying to get that.
That would be great, or at least to refer to it as a a great example of how limited the interspecies.
The other thing is, I agree with Stan completely. We do this with carcinogens all the time. We look for genotoxicity in animal data and epi data, and sometimes you don't have any of these, or sometimes just one. And you can sell the second mechanism, but you have to work and sell it. That epidemiology data has become stronger with time, and now it's into specific classes and specific compounds. So you'll have to make that clear, by analogy with carcinogens, that it's real, and you can regulate it that way. You don't have to have the animal data. You can do it based on the epidemiology if it's strong.
Question? Sorry.
And also, I'm sorry, the zebrafish data supports it too.
Have that too.
Sorry, can I ask a question about the colorful 3-column figure with the ARR, the DNT, DPR, and the US EPA? So first, just a technical question. Point of departure sometimes is a low L, sometimes is a no L?
According to the US EPA definition, actually the point of departure is the point where the extrapolation occurs. So, so meaning actually the dose you actually divided on uncertainty factors. So if you have if the lowest test dose turns out to be a lower, the lowest observed your POD become the no well. And then you divide uncertainty factor to compensate in the absence of a no well. But most of the time,
And
the
that's
point of
usually by a
departure
factor
is
of 10.
equivalent to the no well or the BMD 10. It really depends on how you get it.
Okay, so
It's in the table legend, executive summary table 1. We define the point of departure. This is executive summary table 1, and here is the point of departure definition.
So that sounds like a low L.
I mean, what we've done traditionally is if you only have a low L, then you divide it by 10 and start from there and then you start doing all your other things. Whatever you want to call it, but that's the start.
I see. So when you have a point of departure, can we go back to the very colorful
So
table?
point of departure came fairly recently as a terminology, and it's typically, at least to my knowledge, started being used after the benchmark dose modeling became more and more used in risk assessment. It was usually a low extrapolation or null established as a dose in an animal study. And then, so POD, we would see for benchmark dose modeling for Pbpk derived endpoints. For when it's not, we would traditionally refer to low L, no L, but POD would be the equivalent of a no L.
Okay, so then I think I understand. So the point of departure here in green, it's 10 micrograms per kilogram per day. So in the animal study was 100 micrograms per kilogram per day, and you're dividing that by 10 to go essentially from the low L to the no L, which here is the point of departure. Okay, so I understand that. Thank you. So getting back to Stan's point about uncertainty factors, Andrew was just saying in the intraspecies uncertainty factor for hotspots, they typically consider a factor of 10 for adult variability, but then add an additional factor of 3 to go from adult to child. So I'm wondering then, should that 10 for intra instead be 30?
Wait, you want Andy or you want Oia? You want Oia?
Whoever would like to address this point.
I think you're referring to Andy Salmon when he gave the presentation. Yeah, the other Andy. Yeah. So we made a table as requested, and it shows the 3 columns. And Svalana, you could show that on the screen. Nope. I'll go ahead and start talking. So the first column has the DPR draft THC RBC acetylcholinesterase inhibition with the uncertainty factor 1, 10, 10 with a total of 100. So that's you've seen many times. The column has the Oia finding if the RBC acetylcholinesterase is on screen now is based on the basis for the point of departure. We have the interspecies of 3, which includes the model uncertainty, and then the intraspecies based on the hotspots program, 10 for pharmacokinetic differences, and then a factor of 3, which is actually square root of 10 for pharmacodynamics. So that's where the 30 comes from that you're familiar seeing in the hotspots program. And the additional uncertainty factor is when you're using RBC as the endpoint with a total of 100. And I had to sort of explain is that instead of the 3 is really square root of 10, so that's how come the math comes out to be 1,000. Earlier, you also asked what would we propose the factor for DNT would be. I think we don't have a firm position right now. I think for the intraspecies, it would likely be 10 if it's based on an animal study. We also would like to weight the human studies into the mix before we come up with some definitive answer. Intraspecies, probably 30. That's accounting for pharmacokinetics and pharmacodynamic differences. And here's what the we might need to have additional uncertainty factor, not the FQPA one, but for extrapolating from a low L to a no L or other kind of uncertainty that you take into consideration. Yes.
Hello, I'm Andy Rubin, and I'm a risk assessor with the Human Health Assessment Branch. First, I'd like to say it's somewhat reassuring. I started in this 25 years ago. People used to argue about what the PODs were, and then as time went on, they began arguing more about what the uncertainty were and left the PODs behind. Now we're arguing about both the PODs and the uncertainty factors. So it's a very Talmudic process. Anyway, I just have a couple of brief remarks about the intrahuman or uncertainty factor. Everyone that there is uncertainty regarding the range of sensitivity in human populations. DPR, OIHA, US EPA, the scientific review panel, we all know that there is uncertainty there. The widely adopted default is 1 order of magnitude. In other words, a human uncertainty, intrahuman uncertainty factor of 10. And Dr. Blanc mentioned that this morning that the 10x was is at least traditionally considered a conservative value. Or is traditionally accepted this default value. But in 2008, OIHA issued a technical support document. It's a wonderful document that advocated higher intrahuman uncertainty values. There are lots of examples of chemicals and so forth. So the uncertainty factor for humans is now commonly divided into 2 parts, as we've mentioned, a toxicokinetic part and hope that's not my phone. A toxicokinetic aspect and a toxicodynamic aspect. both a square root of 10, sort of a logarithmic half for each of those components. In the OIHA 2008 document, which was part of their hotspots program, they showed that with respect to the toxicokinetic factor, there's just a whole range. I think they looked at 25 chemicals, and 12 or 13 of them had toxicokinetic factors of 3 or less, actually, and then 5 had toxicokinetic factors of 10 or more, between 3 and 10. So there's a whole spectrum. We can throw all these numbers into the air and then grab the one that we feel has the, that we can support. OEA stated that the reason adopting higher values for the intrahuman, whether we're talking about toxicokinetic or toxicodynamic, is that there are special populations that may have greater sensitivity. So all these results, the spectrum of chemicals, 3, higher than 10, and so forth, emphasize the difficulty in assigning a default uncertainty factor for intrahuman variation. In a way, there's tremendous uncertainty in defining the uncertainty. And I don't know, there's just no right answers about these things. We just have to go to what we feel are best supported now, today. DPR decided for chlorpyrifos, for the present, to stay with an intrahuman uncertainty factor of 10, feeling that adequately represented the human population, the variation in sensitivity in the human population, and obviously there's some disagreement on. There is some modeling work. I think Svetlana might want to comment on this. That with respect to variation in sensitivity with respect to cholinesterase activity, that supports a 10 or perhaps even less. Would any of you like to? Yeah, this is a study that used modeling and bootstrapping, and Lori mentioned it earlier in one form. That's it. That's it. That's it. I'm deferring to them because they have better knowledge of this particular study.
I think it kind of like follow up with what Dr. Rubin just said, that this is an example of a choreasis inhibition, but the table show here is pretty much even if you measure based on the mutual study, and if you do the bootstrapping, meaning actually you keep doing the resampling, and then get the wider range, and to integrate into the Pbpk model. I mean, after you consider all the variation, then you can see that that's the so-called data derived uncertainty factor. This one is kind of like.
Excuse me. We are going to be losing our quorum in a few minutes because we have a plane that's going to. But I think that we're at a point. There are still a lot of things to discuss. We're not going to be able to finish everything today. We're going to have at least one more meeting on this topic, and I'd like to invite everybody back as soon as we get the date picked. In the meantime, it would be very useful, since there is new information that you are considering, if we could get, anything like that distributed to the committee so we can look at it before the next meeting. And then I think we've covered a lot of what was in those charge questions already. And we may be able to, sort of expedite things out at the next session. Stan?
So just quickly, I do think we made a lot of progress today, actually. But I just like to sort of official motion or anything, but just to see, it seems to me the sense of the panel is that the risk assessment should be based on developmental neurotoxicity. And that with the epi used to justify that, but doing the actual dose response on the animals. And so it would be nice, and I keep saying this, I don't think you need a drastic revision of the report, because I don't want you to go off and spend another year doing it. But it would be really helpful if it was possible to just get, and I don't know if it's a matter of doing a track changes on this or just a memo about like taking the stuff you showed us here and putting it down on paper so we can look at it. And then it seems to me, there seem to me two other sort of outstanding issues. One is to finally reach some kind of consensus about the uncertainty factors, and I think there's been a lot of good discussion of that. Another issue that we haven't talked about at all is the exposure stuff, which we'll talk about next time. But I think in terms of the biology, unless somebody here disagrees, I think you've heard a pretty strong consensus out of the panel on where to go with this. Is that? I hope that's helpful. It's all based on what you have in here.
And keep the nerve, the acetylcholinesterase inhibition and that mechanism in.
Yeah. Oh yeah.
Yeah. So you won't have to do that much work.
I have just one other thing to add. I think you really do need to consider the vapor exposure. Concentration is much lower at time of application, but it's this huge reservoir sitting in the field that's then volatilizing over probably days to weeks. So I think you need to at least look at what's the potential exposure to the vapor phase, given that most of the stuff in the field, and then it's volatilizing. So it really should be looked at. How important could that be?
And quickly, I would add, I really would like to see some data on air sampling. That would help to buttress the models. But that's for next time. I understand.
All right, at that point, I'd like to entertain a motion to adjourn. So, moved. Okay, seconded. We're adjourned.
To wherever, to a BART station.